and leaf group demonstrating sex differences in islet transcriptomes in 150 non-diabetic and 30 type 2 donor islets, as well as preprint of small number of single-cell sequencing donor pancreas. These studies identified differences between islet donor traits at a transcript level. Our study assessed a total of 131 donor islets obtained from our collaborators from across the Sydney and Melbourne islet isolation centres. Our aim were to profile transcriptomes and microRNAs from human islets, identify mRNA and microRNA that are associated with age and BMI, and also differentially expressed across sex. And finally, we aimed to identify microRNA regulators that regulate potential differences in islet transcriptome. Our methodology involved assessing de-identified islet samples, isolating RNA, and carrying out bulk RNA sequencing with 88% of the total transcripts detectable, and profiling microRNA where 81% of total microRNAs were detectable. Across these two sets, 52% of the samples had both mRNA and microRNA profiled. All samples passed QC, and data were adjusted for age, sex, and BMI using R, and analyzed were validated using SPSS. We then carried out in silico microRNA target analyses and also some wet lab validation. Of the 63 islets sequenced, there were 38 males and 25 female donors with no significant differences across age or BMI. We first assessed the genome-wide distribution of transcripts that differentially expressed between male and female donor islets after adjustment for age and BMI. Interestingly, we observed that around 2% of the transcripts on each chromosome is significantly different apart from sex chromosome, which showed a slightly higher, around 3% of the transcripts to be significantly different across sex. Interestingly, of the 55 transcripts that had a greater than 1 difference in D-seq normalized value between male and females, 35 transcripts were expressed at a higher level in males, while 20 were higher in females. In our microRNA analysis data set, we had 53 male islets and 48 female islets. Again, no differences between male and female for age and BMI. Interestingly, in our microRNA analysis, there were 32 microRNAs that were associated with age and 47 with BMI, and 10 other microRNAs significantly different between male and females when adjusted for age and BMI. Several microRNAs were common between sex, age, or BMI, while microRNA 199 and 214 were significantly different across sex, age, and BMI. Here we show some of the microRNAs that are significantly different across males and female islet donors. We found that several microRNAs, including microRNA 214, which is known to interact with long non-coding RNA cyst, known for inducing extended activation in females. In addition, microRNA 214 and 189 are expressed on the same cluster, known to target PRDM16, acting as a regulator of fat development and serotonin involved in beta cell survival. In case of all the age-associated microRNAs, I have plotted the first and fourth quartiles for simplicity. Several of these microRNAs were more abundant with age. We then performed target pathway analyses on significant microRNAs, for example, to sex. Using the reactor and pathway analysis, we identified that the sex-related microRNAs were associated with metabolic and translational pathways. Since both microRNA 189A 5P and 214 3P are located on the same cluster, they both are correlative and have potential binding sites to common genes, such as PRDM, 16 associated with fat development, as I mentioned earlier. And fisetin, which encodes 4 ATP-dependent microtubule proteins, is also significantly negatively correlated to both microRNA 1999 and 214. The function of fisetin, however, is not yet known in islets. Fisetin has been suggested to suppress microtubule growth, which is known to provide intracellular trafficking and chromosome segregation. In human islets, microRNA 189 and 214 may potentially bind to fisetin to promote cell regeneration. From the many gene targets associated with age in our analysis, here are some examples of genes associated with age, which include the GLS and NMUR1 genes, which are known to be associated with beta cell function and survival. Similarly, from the gene targets significantly different between sex, we verified that there are genes which were also significantly associated with age or BMI, such as DLK1, CDKN2A with age, and WIF2 and NLRP12 with BMI. DLK1 has been previously shown to be downregulated in islets from type 2 donors, and CDK2N polymorphism through islets and other tissues have been shown previously to impact on diabetes. Here both are downregulated in male and more in males than in females. And finally, I'd like to share some of our recent work obtained from our Danish collaborators from Roskilde University, Denmark, Louise's group. Louise's group first found that overexpression of the age-associated microRNA 219A in INS1E cells significantly reduced the level of insulin transcripts whilst increasing GCG and HES1 abundance. Interestingly, overexpression of microRNA 219A in human islets using lentiviral transduction did not change the basal, but inhibited glucose-stimulated insulin secretion. In summary, I shared some of our analysis for our transcriptomes analysis in 131 human islet preparations. 8 of the 10 sex-associated microRNAs identified are upregulated in females, making it critical for donor-sex balance in islet studies in microRNA analysis. I've also showed in our analysis indicated that several of these microRNA targets are shared by known pancreatic islet transcripts. I then provide you with some examples of dysregulated microRNAs that now help us better explain the findings published earlier, and also provide an example of wet lab validation. Whilst other similar datasets remain more focused on mRNA, our datasets may add value to understanding microRNA regulators that could explain changes observed or reported in pancreatic transcripts. Finally, I'd like to thank my mentor, Anand Hadikar and Mukta Joglika, and the team which I'm very fortunate to be a part of, as well as our collaborators, including Louisa, Wayne, Helen, and Ronald. And also a huge thank you to the JDRF Australia and the Hemsley Trust for the support, and the Flag Trust for specific fundings on RNA-seq, as well as the EASD for giving us the opportunity today to present and share some of this work. Thank you.
Thank you very much, Wilson, for your very interesting presentation. This is an interesting presentation. You have made some correlations between mRNA expression and microRNA expression and gene expression in your samples. This is a very, very important observation. Again, there are no questions from other people present in the room, and also and also no attendees are in this meeting. Anyhow, I like to ask you a question. The correlation that you made are very, very important, and also the validation studies. But maybe I did not understand how many genes, how many transcript messenger RNA are differentially expressed between males and females.
There were several hundreds of genes that were differentially expressed across between male and female islets. It's the 55 that were differentially expressed with a greater than 1 D-seq value, normalized D-seq value, but there was many more than that, so a couple of hundred more.
So you are cut off to describe a gene as differentially expressed, which was, I mean, I guess the p-value was lower than they are. You use an adjusted p-value, right?
Yes. So the values were adjusted for age and BMI. The other,
I mean, the statistical value to see these genes are differentially expressed or not.
Yes, we did adjust it for the false discovery rate for multiple testing, which showed that only a few of the genes were significantly different. But we also performed machine learning, analyzed logistic regression analysis for bootstrapping, which further emphasized that these genes that do show significance are in the bootstrapped selection to validate that these genes were significant.
Okay, I see. Okay, so there are questions from the faculty people in the room. So you have maybe I can make another question. So you have shown some genes that are very important for the beta cell function and for the for the function and for the viability of the islet. Okay, so these genes that you correlate with the microRNA were differentially expressed between male and females?
Yes, so some of these microRNAs were differentially expressed between males and females, and as mentioned earlier, 8 of the 10 were more higher expressed in females compared to males.
Yes, and this is clear, but I'm talking about messenger RNA between males and females. Did you see
yes. Differences?
Yeah, different. You have shown the differences between males and females, but I mean, have you seen genes involved in beta cell function, beta cell survival, or what else?
Yes, so some of the genes, I don't know if I could go back to the slides, but they do show that it was some of the genes are involved with beta cell survival and function, such as I mentioned earlier, GLS glutamine is, which has been shown to be differentially for glutamate production in hormone-secreting cells, and CDK2N, which there's quite a few studies that have shown that it's related to aging, and but also polymorphisms of it has been shown to impact on diabetes as well, through islets and other tinnitus issues.
I see. Thank you very much. Thank you very much. So we can move to the next presentation. That is from Pratek Chawla. The title is A Single Cell Atlas of de novo beta cell regeneration in adult zebrafish identifies hybrid cell states that facilitates diabetes reversal. The work has been performed in Dresden, Germany, in collaboration also with the scientific group in Belgium. So, please.
Hi. Good morning. Thanks, Lorella, for the introduction, and thanks to EASD for giving me the opportunity to present my work. So I'm Pratik, a PhD student in the research group of Professor Nikolai Ninov at the Center for Regenerative Therapies, Dresden. Today I'll be giving you a glimpse of the single-cell atlas of beta cell regeneration that we have generated in the regeneration competent model system zebrafish. So to begin with, we know that of all the endocrine cells, beta cells are one of the prime focus for diabetes research. And although diabetes is a treatable disease, its long-term curative remedy is still not known. From the lost beta cell mass can actually be recovered through multiple sources, which has been shown by various research groups. But what gives rise to a functional beta cell pool is still debatable. So in our group, we make use of the regeneration competent model system zebrafish to study and to uncover the cellular and the molecular mechanisms of endogenous beta cell regeneration. So how do we do so? So we use the NTR-MTZ system, which is for destroying or ablating a particular cell type. What happens? The bacterial nitroreductase is expressed under the promoter of interest, which in our case over here is the insulin promoter. And when it is exposed to the prodrug metronidazole, it is converted to a toxic compound which induces cell death. So as you would be able to appreciate from the image over here, that at 2 dPa, most of the YFP signal is gone because of the MTZ ablation. And as early as 14 dPa, you can see the recovery of the beta cell mass, and which was validated by the glucose measurements that as early as 14 dPa, the blood glucose level returns back to normal. So these are the three aims of my study. First, to characterize the cell types in zebrafish pancreas under homeostasis. Second, to investigate the mechanisms of beta cell regeneration. And third was to compare the zebrafish and the human islet cell types. First, to understand the molecular heterogeneity in zebrafish pancreas, we performed single-cell RNA sequencing from the islet cells from a 2-month-old zebrafish. Downstream analysis showed pretty much all the endocrine and the exocrine cell types, even the ductal cells. When we looked closely into the endocrine population, what we found that there were two populations of somatostatin cells, which we call the delta 1 and the delta 2 cells, and they were transcriptomically very distinct because they expressed, the delta 1 cells express SST1.1 and the delta 2 cells express SST2 genes. These two somatostatin genes, which arise due to genome duplication in teleost fish, are predicted to encode for a full-length and functional somatostatin protein. Second, we looked more closely into the two populations of the islet delta cells, and we found that both the populations are very transcriptomically distinct, with SST1.1 expressing population expressing the Wnt regulator DKK3B, EVNPDX1, and another short polypeptide, the protein YY called PYYB. So they were actually transcriptomically very distinct populations. Interestingly, what we also found apart from this were there was a small population of insulin-positive cells, which were close to 8 to 10% cells, which expressed both insulin as well as SST1.1 gene. So they were actually bihormonal. Apart from these apart from these two genes, the bihormonal population also expressed DKK3B and PYYB. To corroborate our findings from the single-cell RNA-seq data, we made use of the transgenic lines, transgenic reporter lines, to visualize the same, and we could see it at different developmental stages. Here you can see that the SST1.1 cells are EGFP tagged, and the beta cells are in red. So you could see the presence of the bihormonal cells in zebrafish larvae, as well as you could see it in the adult zebrafish. This is a 7-month-old zebrafish islet, and you can see the presence of these bihormonal cells. So green is SST1. Intrigued to identify what is their role in the zebrafish islet. So next. So as in the control group, you can see that there are distinct populations of different islet cell types. So our question was that if we destroy the beta cells, which cell types will show the signs of plasticity and contribute to the recovery of beta cell mass? For this, we again made use of the 10x genomics platform, and we profiled the islet during the entire course of beta cell regeneration, starting from 0 dPa, which is the point of beta cell destruction, 2 and 7 dPa, which are the subsequent hyperglycemic points, and 14 dPa, which shows the attainment of normoglycemia. So as you can see over here is the integrated view of the 4 different time points, 0, 2, 7, and 14 dPa, and the clusters have been annotated with respect to their markers. Interestingly, what we found that all the newly emerging insulin-positive population was coming from the delta 1 population, the 61.1 cells. And you would be able to appreciate it from the Violin plot over here, that the majority of the insulin-positive population is coming from the delta 1 population, and in which the cells co-express both SST1.1 and insulin genes, whereas other cell types show a very relatively minor contribution. And also interesting to see over here that even till 14 dPa, the cells haven't completely resolved to give rise to a monohormonal insulin-positive pool. Next, to further look too closely into this data set, we looked at the insulin expression profile. So basically, I split the data into the insulin expression into low, medium, and high insulin expressing cells based on the beta cell cluster in the control group. So as you can see it over here, at 4 different time points, 0, 2, 7, and 14 dPa, you can see in the delta 1 population, the insulin expression rises from low insulin expressing cell to a high insulin expressing cell at 14 dPa, whereas other cell types continue to retain this low insulin expression profile. Further justifying that the delta 1 cells were the major contributors. Furthermore, we wanted to corroborate our finding that we obtained from the single-cell RNA sequencing dataset. So we performed immunostaining. So as you can see over here, that you can see distinct expression of both insulin and somatostatin antibodies in the control group, and you can see the cells distinctly. As early as 2 days post-ablation, you can see that most of the insulin-positive population is completely wiped out, whereas you can see a distinct expression of somatostatin population. And at 14 dPm, you can see that most of the cells are bihormonal. It's predominantly bihormonal, expressing both somatostatin as well as insulin antibodies. So this further corroborated our finding that both at transcriptomic as well as at the protein level, the cells till 14 dPa are bihormonal and continue to be the chief insulin expressors. Further, we wanted to see that whether these newly found bihormonal cells are functional or not. So we made use of the red calcium indicator. So these experiments were performed in a zebrafish larvae. So we performed live imaging where we used the red calcium indicator KGECO, which was expressed under the insulin promoter, and the SST1.1 EGFP transgenic line. So these 2 markers will be used to basically label the hybrid cells. And we use the insulin flag entry system for ablating the beta cell population. So yeah, as you can see it over here, that yeah, so I have to go a little faster. Yeah, so you could see the response of both the hybrid as well as beta cells in the control islet, as well as in the ablated samples. You could see that the hybrid cells become responsive as early as 4 days post-ablation. So these are the fluorescent traces where you would appreciate that the hybrid cells are responding even at 4 days. And this is over here, actually, you can see the quantification of the area under the curve for single cells. And both the hybrid and the non-hybrid cells under control and 4D updated conditions show a very similar kind of response. But this proves that the hybrid cells are actually functional. The newly found hybrid cells, they do respond to glucose. Finally, we wanted to see if we can induce the formation of these hybrid cells during development without the introduction of injury. So as I showed previously, so we tried to look into the DKK3B gene that is expressed. So as you had seen previously, the delta 1 population predominantly expresses DKK3B as well as other progenitor markers, whereas delta 2 cells do not. And in our beta cell regeneration profile, we could see that when we compare during the entire course of regeneration, you see a slight increase in the DKK3B expression during the course of regeneration. So we made use of 2 systems. First, we constitutively expressed DKK3B gene under the insulin promoter. And we see that overexpression of DKK3B gene increases the proportion of the bihormonal cells. And the second system that we used, just to validate the same thing, we use an inducible system where we express DKK3B under the heat shock promoter. And when we provided a heat shock at 3 and 4 dpf to evaluate it at 5 dpf, we saw very similar results where we see an increase in the proportion of these bihormonal cells, which further shows that DKK3B can induce the formation of the, can increase the proportion of the bihormonal cells without the introduction of injury. Just to conclude my results over here, we found the two distinct populations of delta cells and a small proportion of the beta-delta hybrid cells which are persistent throughout the life. The number of the beta-delta hybrid cells increases during regeneration at the chief insulin expressors. The newly formed beta-delta hybrid cells exhibit a glucose-stimulated calcium influx. And even DKK3B overexpression increases the proportion of the beta-delta hybrid cells. Still, there's an open question that what is the source of these hybrid cells? We believe that it is the delta 1 population, but we still are trying to develop the lineage tracing tools in order to show that eventually the beta cells are giving rise to, eventually the delta 1 population is giving rise to these hybrid cells, which form these mature beta cells. So that's still an open question. And finally, I'm coming to my third aim, where we wanted to assess that how relevant is our study in zebrafish field to the human data sets. So we transcriptomically compared our zebrafish data set with the published human data set. So what we find here very interesting, that the zebrafish delta 2 population is transcriptomically very similar to the human delta cells, whereas the zebrafish delta 1 cells show a partial overlap with the gamma cells, which was very interesting for us. And then we looked closely into the literature. And yes, it actually started making all sense, because there was a paper published which actually showed that gene duplication of the human peptide YY, PYY, generated the pancreatic PPY. So these PYY gene, which is expressed in the human, in the zebrafish delta 1 population, and the PPY gene, which is expressed in the gamma population, according to this paper, are just 10 kb apart from each other. And also, the very two recent papers also show that the PPY cells, the gamma cells, can give rise, can be a contribution for the insulin production once the beta cells have been destroyed. So yeah, so it actually started making all sense, and we really think it is, we still are trying to see if the zebrafish delta 1 cells are the ones which evolved to form the human gamma cells, which is something we are trying to look into. So yeah, I would really like to thank my lab members, especially Professor Nicola Nino, for giving me this opportunity to work on this project, and all the lab members who've really helped me structure up how the project was, the facility for helping me out with all sequencing and microscopy techniques, my TAC members, and definitely all the funding agencies, and the EASD committee for providing me an opportunity to present my work. And finally, we've already released our preprint. So if anybody is interested, could definitely go and check it out. Thanks a lot, and I would have to take more questions.
Thank you, Pratek. Nice, very, very interesting presentation. And yes, actually, you answered to many questions that raised in my mind while you were presenting. But actually, I have still one.
Sure.
So what happens at these kind of delta beta cells? I mean, these hybrid cells, if you look after the 14 days, do they reach the full beta cell phenotype? Did you have the opportunity to look at this or not?
So right now we haven't, but definitely there is another study which shows that, which is a very parallel work which was done by a group from Isabel Moinfroid, and she showed that till 4 months post ablation they do not resolve. So, okay. So they continue to be these bihormonal cells.
Okay. Thank you very much. So we have to move on to next presentation.
Definitely.
That is from Emanuele Bosi from the PISA group. Also, the title of this presentation is an integrated analysis of human pancreatic islet single cells reveals autocrine and paracrine interactions. This paper is in collaboration with Miriam Knopp and Ezio Ziric at the University Libre de Bruxelles. Please, Emanuele.
Hello. So good day. First of all, I'm grateful for this opportunity to present this study in which we sought to identify putative intercellular interactions occurring between the cell types in the human islets. So this study stems from a previous one, which I presented at the last year EASD, in which we analyzed available single-cell data to identify transcriptional signatures of beta cells associated with type 2 diabetes. And okay, so as you know, the communication between islet cells is essential to achieve a timely response to nutrient fluctuations and to regulate the hormone secretion by beta and alpha cells. Although some of these interactions have been characterized quite well, we are lacking an overall picture of all the interactions occurring between the cell types. Latest developments in the field of single-cell data analysis aim to fill this gap. So in this slide, you see a sketch of CellPhoneDB, which is a collection of both data and software for single-cell analysis. And so briefly, the single-cell RNA-seq here is mapped on the CellPhoneDB database, which integrates 3 different databases of interactions that you see here reported. So ligand and receptor interactions, protein complexes, and protein-protein interactions. Then the CellPhoneDB algorithm allows to identify which are the significant interactions occurring between all the possible cell type pairwise combinations. So this kind of approach has already been used to characterize the interactome in the study of tissues other than the endocrine pancreas. And that's generally proved that it's a valuable tool to identify molecular components of importance for the system studied. Therefore, since we had available different single-cell data sets of human islets, we applied such approach in this context. So, as mentioned, we have previously integrated 3 published single-cell data sets of human islets, which allowed us to better highlight signatures of type 2D in beta cells. So this is the work I presented at the last year EASD. And then by analyzing this data set with the CellPhoneDB, we had 2 objectives in mind. So first, to describe the landscape of the human islet interactome, that is to provide a comprehensive description of all the interactions occurring in health between the cell types. And second, to describe how such interactome is altered in type 2D. So to achieve these objectives, we divide the cells according to the diabetes condition of the donor, so ND and type 2D, to obtain 2 subsets. Then we reconstructed the interactome separately for ND and type 2D with CellPhoneDB. We consider a total of 7 cell types that are alpha, beta, delta, PP, ductal, acinar, and stellate cells. And we formally define the interactome as a network in which the nodes represent either the cell types, the ligands, and the receptors. And here, an individual ligand-receptor interaction can be represented as a path in the network, going from the cell expressing the ligand, which we call cell source, to the cell expressing the receptor, which is the cell target. And so this path goes from the source cell type, connects to a ligand, then the ligand interacts with the receptor, and then the receptor connects back to the target cell types. And as an example, you see here the glucagon-glucagon receptor interaction between alpha and beta cells. So since the real interactome network that we obtain embeds thousands of interactions, the resulting object is extremely complex to represent and also to treat. And due to our time limitation, I'm just reporting a subset of interactome, that is the set of the interactions in which the beta cells are involved. So here on the left, you see the total number of interactions between all the pairwise cell type combinations, considering both cell sources and targets. And here on the right, you see the number of interactions in which the beta cells are the cell source, with the interactions with all the cell types. And so remarkably, we found that just considering the total number of interactions, the beta cells have less connections with the other endocrine cells respect to the non-endocrine, with the stellate cells being the most connected ones with the beta cells. Now, this result can be interesting and perhaps maybe also surprising, but one should keep in mind that not all the interactions are equal, in the sense that these can be either weak or strong. So CellPhoneDB provides a measure to quantify the strength of an interactions, which is called the interaction score. And this basically reflects the expression levels of ligands and receptors. So in other words, when the ligands and receptors are expressed at the high levels, the corresponding interaction has a high score. So in this histogram, I reported the distribution of interaction scores of all the beta cell interactions. So as you can see, the distribution displays a so-called power law behavior, meaning that there are most of the connections with low scores, whereas there are few with high scores. So we hypothesized that this metric could be used to prioritize important interactions for beta cells. Thus, we ranked interactions according to this score, and we went to look at the highest score interactions. And actually, we found that among the top 40 interactions, there are the important ones, in the sense that all of these include the insulin as a ligand. So basically, the interaction insulin receptor between beta cells and all the other cell types, and interaction insulin and insulin degrading enzyme. And the other strongest interactions involve the islet amyloid polypeptide precursor, or amylin, which is a molecule that is co-secreted with insulin. And this ligand interacts with the insulin degrading enzyme. Now, looking at the interactions in which the beta cells are expressing the receptors, so they are acting as cell target, we see that the highest score interactions include important interactions with the endocrine cells, such as glucagon with alpha cells and the somatostatin with delta cells. So this provided a sort of validation of our model, in the sense that it is able to recapitulate known fundamental interaction as the most important ones. So now we pass to the second connectivity. Therefore, the type 2D conditions seem to favor the emergence of new interactions that were not present in the ND interactome. And again, we focused on the changes that were specific to beta cells, revealing a considerable number of alterations, with almost 600 new interactions emerging in type 2D, and 404 being lost. So we again ranked these changing interactions according to their score, and evaluated if they contained signatures that could make sense in the context of type 2D. So an example I want to share is the emergence of new FA-FNA interactions between delta cells, beta cells, and ductal, acinar, and stellate cells. So in particular, beta cells express FNA5, which normally interacts with other beta cells FNA receptors to properly regulate insulin secretion in response to glucose levels. So therefore, what we found is the emergence of new aspecific interactions, which is, of course, interesting. Although deserves more thorough examination. And then, looking at the lost interactions, we found Yes.
In terms
yes.
of interaction, you have shown that there is a difference in the number, but I mean, in the graph, you have shown the graph in a non-diabetic subject in which we have beta cells and other cells, and you arrive to the stellate cells. Have you seen any? Okay, please.
So yes, of course, we have a very high number of potential interactions to look into, and of potential relevance, which I think I showed you with the changing interactions. So we can also explore in the terms of genes, so genes involved in these interactions, which are associated with biological themes that are usually interesting when studying at beta cells or diabetes, such as inflammation or the transition to the differentiation. So, for instance, the stellate cells, which are interesting. Also, looking into the literature, they are understudied, but they seem promising. We found, for instance, some interactions also involved in the inflammation in type 2. Okay. So.
Okay. Thank you very much for this. Just about the stellate cells, how they do participate in this interaction? Much with the ligands or with receptors?
So, for instance, we found the stellate cells producing the neuroleptin-2, which is a regulator, for instance, of that has been found associated with the inflammation. In this sense, they are the sources, but they have a lot of interactions also as targets. So they express also receptors.
The receptor, okay. Thank you very much. Thank you very much. Now we can move to the next presentation, that is from Amaya Lopez Pascual. The title of the presentation is "The transcription factor CEBPG regulates beta cell function," and the study has been done in Sweden.
Thank you for the opportunity to present the study "The transcription factor CEBPG regulates beta cell function," carried out during my postdoc at Lund University Diabetes Center. Impaired beta cell function is central for the development of type 2 diabetes. However, the underlying molecular mechanisms have been hard to determine due to the complexity of the pancreatic islets. With our new algorithm for the single-cell RNA-seq data we have performed in human islets, we want to achieve a comprehensive analysis of how biological processes are affected in type 2 diabetes. Thanks to this approach, we identified type 2 diabetes affected gene regulatory networks in beta cells. I would like to briefly explain the endoplasmic reticulum stress and the unfolded protein response pathways in beta cells. Among other triggering factors, increased proinsulin synthesis is a major direct cause of physiological ER stress in beta cells. misfolded proinsulin in the ER binds to and titrates away the molecular chaperone BIP from the UPR sensors that are 3 ER transmembrane proteins, PERK, 81, and ATF6. Activated PERK phosphorylates EIF2 alpha, which generally inhibits all translation. However, phospho-EIF2 alpha activates ATF4 translation. 81 splices XBP1, promoting its translation. Finally, ATF6 translocates to Golgi apparatus for proteolytic processing. The translocation of the transcription factors ATF4, XBP1S, and ATF6P50 into the nucleus activates stress response genes that play important roles in mitigating the ER stress initially, but could lead to the cell death with chronic ER stress. Since CBPG was found to be highly ranked in a UPR cluster in human pancreatic beta cells, we aim to investigate its role as a potential novel beta cell regulator. CCAAT enhancer binding protein gamma, CBPG, is a truncated isoform of the CBP family of transcription factors that does not transactivate by itself but heterodimerize with other family members. Also, some authors describe that CBPG has a role in the regulation of protein production at the stage between post-translational modification and protein secretion. So, we hypothesize that CBPG participates in transcriptional regulation and stress response. Here, we aim to investigate the regulatory and biological function of CBPG in pancreatic beta cells. We use the INS1 A3213 rat insulinoma cell line as a model of beta cells and CBPG siRNA for functional analysis such as qPCR, glucose-stimulated insulin secretion, and immunofluorescence. We also use human islets for functional validation with CBPG siRNA. Also, we have done a transcriptomic analysis from a CBPG knockdown in rat INS1 beta cells and a chromatin immunoprecipitation and sequencing analysis in rat INS1 cells, simulating hyperglycemia by chronic treatment in glucotoxicity. So far, the results showed that siRNA-mediated CBPG silencing of INS1 cells result in increased insulin mRNA expression. In human islets, CBPG silencing increased insulin expression and decreased UPR markers expression. Glucose-stimulated insulin secretion increased after CBPG knockdown in INS1 cells stimulated with different secretagogues. CBPG knockdown increased basal insulin secretion in human islets. We also found that 24-hour treatment with tapsigargin, an ER stressor, increased CBPG and UPR markers ATF6, ATF4, and XBP1S expression, and decreased insulin expression. High glucose decreased CBPG and insulin expression. Thus, CBPG knockdown suppressed the effect of tapsigargin on glucotoxicity treatments on insulin and UPR markers. This data suggests that CBPG could have insulinotropic properties and mediate the unfolded protein response to endoplasmic reticulum stress. We performed ChIP-seq analysis to identify CBPG direct target genes in INS1 beta cells, cultured either in control or glucotoxicity for 4 days. KEGG pathway analysis of CBPG-bound genes in promoter regions of INS1 cells in control group indicates an enrichment in insulin secretion pathway, while glucotoxicity conditions led to the regulation of genes associated to diabetes. Thus, CBPG could be involved in the regulation of beta cell stress response. We further integrated the ChIP-seq-based CBPG target genes and CBPG knockdown responsive genes. Overlapping CBPG bound and knockdown differentially expressed genes were submitted to KEGG pathway analysis, which pointed to pancreatic secretion, insulin resistance, and signaling pathways. Later, we identified up and down regulated genes exclusively present in control or leukotoxicity conditions. Hence, CBPG has a role in beta cell function, probably through insulin signaling, pancreatic secretion, and insulin-resistant pathways. To conclude, CBPG is a novel regulator of beta cell function genes with insulinotropic properties. CBPG is needed for unfolded protein response to endoplasmic reticulum stress, showing that the prediction from our single-cell RNA-seq algorithm is correct. Finally, our study provides the first genome-wide identification of CBPG target genes in beta cells, proving an important role in beta cell function. This work has been carried out with my colleagues at Wirup Lab and Karolinska Institute, and the collaboration of members at Lund University Diabetes Center, ExoDiab, and Skånes Hospital, and thanks to the supporting organization. Thank you for your attention.
Thank you very much, Amaya, for this very interesting presentation. So you started from your study in islet cells from type 2 diabetic subjects, and you arrived to validate the function of these genes in a beta cell model, the INS1 cells, and also you performed your study in the human islets. So, very well done.
Thank you.
Sorry, do you hear me?
Yeah.
Okay. Work very well done. So, my question is this: do you think there is the possibility to modulate the expression of this gene from a pharmacological point of view? This is just a question regarding what you think about it.
I think I haven't checked, to be honest, but I think it's not possible so far because it's a really new protein. It's not linked to many diseases, so I think so far there's no much interest on targeting this protein. So until we discover a little bit more, there will be no targeting drugs for this. Okay. But it will be very interesting to check if there's any possibility of blocking only this factor and not the others in the family.
Sure, and I think that the very interesting part of this work that I guess it is in progress, right? Because you have seen that there are these protein targets many genes involved in the beta cell function.
Yeah.
Right.
Mm-hmm. Yeah, I think it's involved in a lot of beta cell function genes. Yeah.
Are you exploring these target genes? Just a curiosity.
No, for the moment we haven't done any more functional analysis on the genes, but we will do a little bit more deep understanding of the functions.
Okay. Thank you. Thank you very much. So if there are no questions, and actually there are not, I think that with 3 minutes we would have 3 minutes to continue the discussion if someone can ask question.
I'm seeing in the questions and answers that there are some. Okay.
They are not, right?
So Miriam Knopp says that, if I read the slide correctly, CBPG silencing did not affect basal UPR in in-swan cells, but did in humans. Any explanations for this discrepancy? Yes, I think this is a really good question. We haven't checked the expression of CBPG in the other cell types in islets, and could be that CBPG is also controlling other UPR markers in the other cell types. But also I think that could be something that is not directly related to the expression of these genes that we measure, but the action of the cascades. So maybe we haven't explored the full view of the UPR in the INS cells to figure out what's happening also in beta cells. And Heinrich Mulder also says that if there are any genetic variants in CBPG genes associated to type 2 diabetes or any metabolic traits, we checked that. I think we checked that, yes. And there were some genetic variants, but not associated to type 2 diabetes. And I think there were some metabolic traits or other comorbidities associated to type 2 diabetes that have some genetic variants in CBPG, yes.
Okay, I apologize with all the speaker and with all the people in the room, the audience, because actually I do not see the questions that are typed, and I don't see them now, and I haven't seen them before if we got some of them, so I apologize about it, and actually I don't see people in the room, so I guess there is some technical problem about it. So I have following the instruction from the technical support, but I cannot see. I don't see any. Can the technical support have the command of this problem and allow me to Okay, and for the question that have not been answered before from the audience, if there were any one, I can send them to the speakers so that they can reply directly to the attendees. So we can move to the last presentation, that is from Itziar González Moro from the university from Spain.
Hello everybody, my name is Itziar González Moro and I'm a PhD student at the University of the Basque Country. Firstly, I want to thank the organizers for inviting me to give this talk. And today I am presenting you the results of a project we are performing in collaboration with the Girona Biomedical Research Institute. This work is focused on the functional characterization of L-non-choline RNA that is codified from the first intron of the transglutaminase gene. Transglutaminase 2 gene codifies a very ubiquitous protein with the same name. This enzyme is able to trigger a wide variety of functions, among which we can highlight its GTPase function participating in the intracellular signaling mediated by G proteins, also its protein kinase activity, or even its implication on cell apoptosis. Due to this diversity of localization and functions, transglutaminase 2 is associated with different diseases, including celiac disease, where anti-transglutaminase antibodies are present, cancer, fibrosis, but also diabetes, which we are interested in. Focusing on diabetes, in an article of 2002 demonstrated that knockout mice for transglutaminase 2 have higher blood glucose levels than control mice after an oral glucose stimulation. Moreover, insulin levels were decreased in those knockout mice, as you see on the image in the middle. And moving to the last figure, they show how a TGM2 knockout mice pancreatic islets, the insulin released after glucose treatment is also minor compared to transglutaminase 2 producing islets. In this article of 2018, it was described there is a long-non-coding RNA of 1,000 base pairs that is codified from the first intron of the transglutaminase 2 coding gene. They described the expression of this long-non-coding RNA correlated with the expression of the coding gene TGM2 in many human tissue samples. Moreover, you can see marked in green, this long-non-coding RNA has a polymorphism in the first exon, which can alter its structure and also its function. So our objective is to analyze the possible implication of this long-non-coding RNA that we have called long-non-coding TGM2 in the pancreatic beta cells and in the development of type 2 diabetes. We decided to characterize the function of long-non-coding TGM2 working with a pancreatic beta cell line called endocyte beta H1. Firstly, we compared its expression on a variety of human tissue samples and show with black bars it is a very ubiquitous long-non-coding RNA and moreover present in very different quantities. What is interesting for us is that its expression is fairly good correlated with the one of the coding gene TGM2, illustrated with red spots. On the right, I show you this long-non-coding RNA is located in the nuclei of the pancreatic beta cells, which indicate us a possible implication in the transcriptional regulation. The next step was to perform long-non-coding TGM2 overexpression and silencing experiments on beta cells and check if the correlation between the long-non-coding and the coding TGM2 gene is maintained in beta cells. On both experiments, the coding gene expression was in line with the pattern of the non-coding gene. In summary, overexpression of long-non-coding TGM2 provokes an increment in TGM2 coding expression, and a silencing experiment results in TGM2 diminish. Moving to a better acknowledgement of the SNP, there are some meta-analyses and genome-wide studies that have found a genetic association between the genotype and the risk to develop type 2 diabetes. For this reason, and in collaboration with Dr. Paco Ortega and José Manuel Fernández from the Girona Biomedical Research Institute in Catalonia, we analyzed the genotype of this SNP in a cohort of 557 people in which obese were overrepresented. You can see in the table on the top, the homozygous genotype for the risk allele is overrepresented in people with diabetes. Moreover, we observed that in women there was an association between the homozygous genotype for the risk allele and some type 2 diabetes-related parameters, as high blood glucose and elevated insulin resistance index. In addition, they also presented higher levels of insulin both in basal and after glucose stimulation. The next step was to understand the effect of the SNP in the long-non-coding TGM2 itself. In the figure on the left, you have an in silico secondary structure prediction of the long-non-coding RNA with the protection allele on the left and with the risk allele on the right, where differences in the structure are visible. To check whether this in silico prediction was accurate, we performed an in vitro transcription of the long-non-coding RNA harboring the risk allele or the protective allele, and we ran an electrophoresis mobility assay. As you see on the image, the electrophoretic pattern was different, so we can conclude that the polymorphism has an impact on its secondary structure. After observing long-non-coding TGM2 regulated TGM2 gene expression in beta cells, we next performed some experiments to clarify the impact of its allele on the transcription and regulation of TGM2. To this aim, we upregulated long-non-coding TGM2 using allele-specific overexpression plasmids in beta cells. As observed in these two graphs, when long-encoding TGM2 harbored the risk A allele, the upregulation of TGM2 was higher compared to the protective G allele, both in RNA and at the protein level. We decided to go a step forward and analyze the TGM2 promoted activity in the presence of long-encoding TGM2. On this schema, I show you a summary of the experiment. We designed a plasmid in which the luciferase gene transcription was dependent on the promoter of transglutaminase 2. The idea was to overexpress the long-encoding TGM2 with either one allele or the other and check the levels of luciferase on each condition. Going to the results, long-encoding TGM2 upregulation increases transglutaminase 2 promoted activity significantly compared to control cells. And additionally, the risk allele harboring long-encoding TGM2 have even higher implication on this promoted activation. In summary, our preliminary data suggests long-encoding TGM2 is able to regulate transglutaminase 2 coding gene in pancreatic beta cells. And moreover, the polymorphism present in the first exon of the long-encoding RNA not only changes its secondary structure, but also exacerbated the effect of the long-encoding TGM2 in the regulation of the transglutaminase coding gene, suggesting an implication on the development of type 2 diabetes. Thank you very much for your attention, and any questions, suggestions, or comments are very welcome. Thank you.
Thank you very much, Izia. So I am kindly ask you to check if there are questions for your presentation, because I'm not still able to see them, and I would very much appreciate if you can see and read them and answer.
No, I can't see them.
Okay. Okay, so I have a question. You have shown that the SNP of the is correlates with the OMA-ER, right? In the population that you studied. Would you please speculate about it? I mean, which is the function of these genes, TGM2?
Well,
I guess that if it is involved in the OMA-ER, it affects the OMA-ER, maybe as a role in the peripheral tissues, right?
Yes, we are working now with the pancreatic beta cell line endoC-beta-A2, but it is true that this long-encoding RNA may be implicated in the insulin secretion, and now we are performing some experiments to analyze that. And this insulin secretion could also have effects in those peripheral islet and peripheral cells. So now we are performing experiments to analyze so.
Okay. Okay, so could you please check if there are questions, and the technical support can see if there are questions, maybe. And
question of Mirang Knop.
Okay, please.
Yes, she answered I could please clarify in which cells I did perform the knockout and the overexpression studies. I have to say that all these experiments are performed in the endoC-beta-1 cells.
Thank you, Miriam, for your questions. Okay, so I guess if there are no other questions, we can close the session. I like to thank you all the speakers for their presentation, this very interesting presentation. that I'm sure that we'll have, you can publish, and that they will have a future. They can give us, they are teaching us many things. And I thank all the attendees, if there are any in the room. So I'm going to check. I'm going to check if there were any questions during the session that I can send. We can send with the help of the technical support. We can send to the speaker. Thanks again, and enjoy the EASD meeting. And okay, good. Enjoy the meeting. Goodbye, and I hope to see you soon in another occasion. Thank you very much again.
And with the ongoing COVID-19 video series, we were able to respond to a fast-changing information landscape, sharing vital information on diabetes and the pandemic as and when it arose. 2021 being insulin's centenary year, we have marked the occasion with a series of 100 insulin-related features, ranging from the history of insulin to the insulin replacement therapies of the future. Also new this year is the long and the short of it, in which eminent experts interview one another on a key aspect of diabetes treatment or research. Finally, there is the patient who changed the way I think about diabetes, our podcast series in which leading diabetes clinicians describe the cases that have shaped their vision and understanding of diabetes care. We have plenty more exciting new initiatives ready for release, so do keep a lookout on horizons over the coming months for, amongst others, the briefing room, 5 things we don't yet know, and insulin in my country. Meanwhile, of course, our core roster of courses keeps growing month on month. Just this week at the EASD's second virtual annual meeting, we will be launching 5 new modules covering the treatment of NAFLD, painful diabetic neuropathy and fear of hypoglycemia, plus new courses on obesity and diabetes and on real-world evidence. Further courses, including beta cell physiology, lifestyle interventions, structured education, and of course insulin, are in development for 2022. Over the last year, with support from our authors, expert reviewers, assessment setters, and several of the EASD's study groups, we have worked hard to improve the assessment aspect of our modules and increase their focus on practical clinical scenarios. 40 academic reviewers are now set up on the site to support its content development. And we have a new messaging and feedback system launched in May to help us communicate better with users and potential users of the e-learning platform. All of this work has helped us to achieve one of the platform's principal goals for 2021. It was clear from user feedback over the last couple of years that learners really want to be able to earn continuous professional development (CPD) points from doing our courses. This summer we submitted our first module for CPD accreditation by the UK's Royal College of General Practitioners. We're delighted to announce now that our submission was a success, and so far one of our modules has been given CPD accreditation, with another 6 in the process of receiving accreditation. The entire site is also being submitted for accreditation by the Royal College of Physicians in October, and plans are underway to submit our online courses to national accrediting bodies around the world. So why not create your free account today? Join us and a host of well-known faces from the world of diabetes research, treatment and management, and develop your professional learning alongside the experts. Since its launch in 2018, the EASD's e-learning platform has expanded at a remarkable rate in its scale and reach. This year in particular has seen it mature into an established resource of systematically reviewed, clinically focused education for the whole diabetes care team. We now offer more than 70 modules, each one carefully crafted with and presented by leading experts from around the world, with course topics ranging from therapeutic inertia and phenotypic variability to metabolic surgery and islet cell transplantation. The site is visited by an average of 2,300 visitors every month and has an audience that is truly international, hailing from every inhabited continent on the globe. We also reach a broader healthcare world via social media and are building an enthusiastic community of followers, currently standing at over 2,700 on Twitter. This year saw the launch of a new section on the EASD e-learning platform, Horizons. In this section, we provide visitors with weekly editorial updates on diabetes news from the conferences and journals, alongside our range of filmed initiatives. Horizons is now home to over 100 educational films, looking at the latest innovations and debates in diabetes research and treatment. Last year, our series Around the Diabetes World in 80 Days brought together specialists from around the world to highlight the particular challenges of diabetes care in their region. And with the ongoing COVID-19 video series, we were able to respond to a fast-changing information landscape, sharing vital information on diabetes and the pandemic as and when it arose. 2021 being insulin's centenary year, we have marked the occasion with a series of 100 insulin-related features, ranging from the history of insulin to the insulin replacement therapies of the future. Also new this year is the long and the short of it, in which eminent experts interview one another on a key aspect of diabetes treatment or research. Finally, there is the patient who changed the way I think about diabetes, our podcast series in which leading diabetes clinicians describe the cases that have shaped their vision and understanding of diabetes care. We have plenty more exciting new initiatives ready for release, so do keep a lookout on Horizons over the coming months for, amongst others, the briefing room, 5 things we don't yet know, and insulin in my country. Meanwhile, of course, our core roster of courses keeps growing month on month. Just this week at the EASD's second virtual annual meeting, we will be launching 5 new modules covering the treatment of NAFLD, painful diabetic neuropathy and fear of hypoglycemia, plus new courses on obesity and diabetes and on real-world evidence. Further courses, including beta cell physiology, lifestyle interventions, structured education, and of course insulin, are in development for 2022. Over the last year, with support from our authors, expert reviewers, assessment setters, and several of the EASD's study groups, we have worked hard to improve the assessment aspect of our modules and increase their focus on practical clinical scenarios. 40 academic reviewers are now set up on the site to support its content development. And we have a new messaging and feedback system launched in May to help us communicate better with users and potential users of the e-learning platform. All of this work has helped us to achieve one of the platform's principal goals for 2021. It was clear from user feedback over the last couple of years that learners really want to be able to earn continuous professional development (CPD) points from doing our courses. This summer we submitted our first module for CPD accreditation by the UK's Royal College of General Practitioners. We're delighted to announce now that our submission was a success, and so far one of our modules has been given CPD accreditation, with another 6 in the process of receiving accreditation. The entire site is also being submitted for accreditation by the Royal College of Physicians in October, and plans are underway to submit our online courses to national accrediting bodies around the world. So why not create your free account today? Join us and a host of well-known faces from the world of diabetes research, treatment and management, and develop your professional learning alongside the experts.
Tell me something, girl. Are you happy in this modern world? Or do you need more? Is there something else you're searching for? I'm falling. In all the good times I find myself longing for change.
Aren't you tired trying to fill that void? Or do you need more? Ain't it hard keeping it so hardcore? I'm falling. And in the good times I find myself longing for a change. And in the bad times I fear myself. I'm off the deep end, watch as I dive in. I'll never meet the ground. Crash to the surface where they can't hurt us. We're far from the shallow now. In the shallow, shallow. In the shallow, shallow. In the shallow, shallow. We're far from the shallow I'm off the deep end, watch as I dive in. I'll never meet the ground. Crash to the surface where they can't hurt us. We're far from the shallow now. In the shallow, shallow. In the motherfucking shallow. In the shallow, shallow. We're far from the shallow now.
Uptown girl, she's been living in her uptown world. I bet she's never had a backstreet guy. I bet her mama never told her why. I'm gonna try for an uptown girl. She's been living in her white bread world as long as anyone with hot blood can. Now she's looking for a downtown man. That's what I am. And when she knows what she wants from her time, and when she wakes up and makes up her mind, she'll see I'm not so tough just because I'm in love with an uptown girl. You know, you seen her in her uptown world. She's getting tired of her high-class toys and all the presents from her uptown boys. She's got a choice. Uptown girl, you know I can't afford to buy her pearls. Or maybe someday when my ship comes in, she'll understand what kind of guy I've been. And then I win. And when she's walking, she's looking so fine. And when she's talking, she'll say that she's mine. She'll see I'm not so tough just because I'm in love.
Welcome to my presentation entitled Leukocyte Counts and T-cell Frequencies Differ Between Novel Subgroups of Diabetes. I'm happy to present to you today these novel results of the German diabetes study, which have been recently accepted for publication in Diabetes. Immune cells play a very important role in the pathogenesis of type 1 and type 2 diabetes, and also in diabetes-associated complications, such as cardiovascular disease and neuropathy. Frequencies of circulating immune cells associate with Recently, the classification into type 1 and type 2 diabetes has been challenged by the definition of novel diabetes subgroups based on metabolic parameters. Patients positive for glutamic acid decarboxylase autoantibodies are defined as severe autoimmune diabetes. Based on cluster analysis including age at diagnosis, BMI, HbA1c, and HOMA2 values for beta cell function and insulin resistance, 4 other types of diabetes are defined: severe insulin-deficient diabetes, severe insulin-resistant diabetes, mild obesity-related diabetes, and mild age-related diabetes. These subgroups do not only differ in metabolic parameters, but also in risks of diabetes complications. The SID subgroup, for example, has a high risk for retinopathy. The ZIRD subgroup has a high risk for nephropathy, liver fibrosis, and cardiovascular disease. Furthermore, our group could recently show that different subgroups differ in their circulating levels of biomarkers of inflammation. Here you see as an example the pro-inflammatory cytokine interleukin-6, with very high levels in the ZIRD subgroup and lower levels in the SIDE subgroup. Therefore, we were interested in the question whether circulating immune cell profiles are different between novel subgroups of diabetes and whether these differences are explained by the clinical variables used to define the subgroups, so age, BMI, HbA1c, HOMA2 values, and GADA. For our study, we used the German diabetes study. This is a prospective longitudinal study including patients from 18 to 69 years that have been recently diagnosed with diabetes. The participants are intensively phenotyped with imaging techniques, anthropometric measures, and metabolic measurements, and followed up for 15 years. From all GDS participants recruited at the German Diabetes Center, we had subgroup annotation available for around 1,000 participants. From these, we had complete data for leukocyte counts from 669 patients and flow cytometry with more detailed information on immune cells available for 201 participants. Automated white blood cell counts were performed on a Sysmex KX21 or on an XP300. Flow cytometry analysis of fresh whole blood samples was performed on a Faxcalibur. Pairwise differences between cell types across diabetes subgroups were estimated with linear models after log transformation of data, and p-values were adjusted for pairwise multiple comparisons by Tukey-Kramer. Correlations were estimated using Spearman's rank correlation coefficient. Here you see the participant characteristics. All SIDE patients were GADA positive. The SID patients had a high HbA1c and low HOMA2b values. The ZIRD subgroup has a high BMI, high insulin resistance values, and a high triglyceride level. The MOD subgroup is characterized by a high BMI and the MARD subgroup by a high age. Here we see the leukocyte counts in the blood in the different subgroups. We see that ZIRD and MOD have the highest leukocyte levels, higher than in MARD and higher than in SIDE. Also, the leukocyte composition differs between the subgroups. The SIDE subgroup has the highest percentage of lymphocytes, and this is significantly different from ZIRD, MOD, and MARD. Neutrophil frequencies were higher in MOD than in SIDE, and consequently the neutrophil to lymphocyte ratio was higher in ZIRD, MOD, and MARD compared to SIDE. We looked for correlations between the leukocyte frequencies and the different metabolic parameters, and as you see here, the leukocyte numbers correlated positively with age, BMI, HbA1c, HOMA2 values, and negatively with GADA. The direction of the correlations was similar for neutrophils and the neutrophil to lymphocyte ratio, and the direction was opposite for the lymphocyte frequencies. When we adjusted the analysis for the variables used to define the subgroups, differences in leukocyte counts between groups were attenuated. So a large part of the differences between subgroups can be explained by the clinical variables. CD4 T cell frequencies were higher in SEERT compared to ZITE, MOD, and MARD, and CD8 T cell frequencies were higher in ZITE and MOD compared to SEERT. The CD4 to CD8 T cell ratio was higher in SEERT compared to ZITE, MOD, and MARD. We also looked at regulatory T cells, and I here show results for the CCR4 positive Tregs. These are antigen-primed regulatory T cells with immediate suppressive function. These cells can be recruited to various metabolic tissues, for instance gut, adipose tissue, and pancreas, but their role in diabetes is mainly unexplored. We saw that circulating CCR4 positive Tregs had higher frequencies in SEERT compared to ZITE and MOD, and higher frequencies in MARD compared to ZITE. When we here looked at the correlations with the metabolic parameters, we saw that CD8 T cell frequencies were negatively correlated with age, BMI, HOMA2 values, and positively with GADA. For CD4 T cells, the CD4 to CD8 T cell ratio and the CCR4 positive Tregs, the correlation was in the opposite direction. Also here, after adjusting for the variables used to define the subgroups, differences in leukocyte counts between groups were attenuated. To conclude, diabetes subgroups differ in leukocyte counts and circulating T cell frequencies, namely CD4 plus and CD8 plus T cells and CCR4 positive regulatory T cells. Diabetes subgroups do not differ in frequencies of B cells, NK cells, NKT cells, and total regulatory T cells, which I did not show here. The SEERT subgroup is characterized by high leukocyte counts, the highest neutrophil to lymphocyte ratio, and a high CD4 to CD8 T cell ratio. Differences in immune cell counts and frequencies are partially explained by clustering variables. And overall, we think that the different immune cell frequencies in diabetes subgroups suggest distinct inflammatory processes in the different subgroups. With that, I would like to thank everyone who participated in the study, and I would like to thank you for your attention. Hi everyone, and welcome to the discussion. Since we don't have a chair in our session because he didn't appear, I will be answering your questions if they come in. And I don't see any questions now yet, but please put your questions, and I would be happy to answer them. In the meantime, maybe I can promote a bit the publication that was just put online at Diabetes, and there we also have some more data on correlations of the different cell types that we analyzed with the markers in serum. So we also analyzed the correlation of the different cell subsets with biomarkers of inflammation that we measured in the serum of the patients. And these data I didn't show here, but there we see, for example, very interesting correlations in the MART subgroup with TREAK and GINA that negatively correlate with the neutrophil to lymphocyte ratio, and also some correlations with the CCR4 positive Tregs with IL-15RA. And yeah, this might also be very interesting candidates for follow-up studies. So I still don't see any questions from the audience. Maybe there are some questions from the other participants in the session. So I think it's a bit weird if I would ask myself questions. So because there are no questions from the audience, and I'm not sure if we should then proceed with the next presentation.
Start with my presentation. Okay, so the title of my project is Visceral Adipose Tissue Derived PD-1 Positive CD40 Conventional Cells from obese patients with type 2 diabetes are pro-inflammatory cells. To give you just a brief introduction. And it is well known that obesity comes with regular saturation of adipose tissue. We have inflammation, we have... Can I go on? Right. We have hypertrophy of adipocyte, and also we have loss of insulin sensitivity. And due to the inflammatory aspect of this disease, a lot of attention has been focused on macrophages, but there are some evidence that support also a role for less obvious candidates in a context that is not antigen specific, like such as adaptive immune cell. It has been demonstrated that CD8 T cells in obese mice infiltrate fat tissue and promote macrophage infiltration. The frequency in human of type 1 helper T cells from the visceral adipose tissue correlate with insulin resistance. And our group demonstrated that T cells isolated from the fat tissue of beta diabetes are resistant to suppression induced by TGF-beta. And even there has been some profiling of the T cells in type 2 diabetes, but there is a lot of uncertainties about their role in type 2 diabetes. And in particular, we wonder if there are a specific subpopulation that may be linked with type 2 diabetes. And we therefore set up a study to gain insight of T cell profile in the visceral adipose tissue of obese patient undergoing bariatric surgery. And the patient have been divided into groups, those without diabetes and those with pre-diabetes or type 2 diabetes according to the level of glycemia. And the visceral adipose tissues has been processed to isolate stromal vascular fraction, which contains T cells. And we started by setting up a flow cytometry panel to investigate the phenotype of these cells. And once identified a population, we proceed with RNA sequencing and TCR sequencing. And as you can see by our unsupervised analysis of flow cytometry data, there is a cluster of conventional CD40 cells with an effector memory phenotype that is Ki67 negative, so non-proliferating, and with a positive expression of PD-1 that is reduced in the group of diabetic patients.
And also by supervised analysis, we could see that the number of PD-1 positive CD40 conventional cells is reduced in this group. And so the next question was about the profile of these cells. So we sorted the PD-1 positive and PD-1 negative cells and performed RNA sequencing. And as you can see in the unsupervised PC analysis, I mean, if you look at PC1 and PC2, you couldn't see any obvious factor. But if you look at PCR3, you can see that cells from diabetic group cluster separately from those then of non-diabetic group. And we found that genes that are upregulating PD-1 positive cells are related to inflammation, inflammatory response, leukocyte migration, and lipid metabolism. Moreover, the gene set enrichment analysis showed that PD-1 positive cells from diabetic patients are enriched for genes related to inflammatory response and TNF-alpha signaling via NF-κB. And we then decided to take a closer look to the PD-1 positive cells from diabetic patients. As you can see that the majority of PD-1 positive cells have an effector memory type in comparison to the PD-1 negative, and also they are more proliferative than their negative counterpart. They produce more coenzyme B, and when stimulated with the PMA ionomycin, they tend to produce more interferon gamma and TNF-alpha cytokines. But then instead, when we look at, we compare PD-1 positive cells we found with from the non-diabetic patients, we couldn't see any differences. Despite the RNA sequencing suggests that PD-1 cells from diabetic patients should be more pro-inflammatory. And then we interrogated us if antigen specificity is relevant to this to type 2 diabetes, and if PD-1 cells from diabetic patients can undergo clonal expansions. So to answer this question, we sequence the TCR, the most variable part of TCR receptor, which is a beta chain of CDR3 receptor. And for this analysis, we also included subjects that are lean and non-diabetic. And we also include T cells from the peripheral blood from all the type of patients. And this is a very preliminary result, but there is something neat. You can see that in the link control subjects, both in the PD-1 positive and PD-1 negative cells, if you look at inverse Simpson index, that is a measure of clonal expansion, you can see the TCR of link control subjects is clonally expanded in the tissue compared to peripheral blood. And this trend is not observed in obese patients. This suggests that the obesity may alter the TCR repertoire. And then we made a clonal tracking analysis in which each subject has been evaluated for the presence of clones. We counted the number of clones that are shared between the visceral adipose tissue and the peripheral blood. And these shared clones are those identified as recirculating one that can migrate from the tissue to the periphery or vice versa. And the interesting point here is the trend that we observe only in diabetic patients. If you look at the PD-1 positive cell from diabetic patients, you can see there's a higher number of recirculating clones in PD-1 positive and this decrease in PD-1 negative. And this trend is unique to diabetic patients, is not evident neither in link control or non-diabetic subject. And we have our RNA data that support these findings, because the genes related to the residency of T cells are those that are upregulated in non-diabetic subject and not in diabetic one. So in conclusion, the PD-1 cells, CD40 conventional cells that are localized in the visceral adipose tissue of obese patients with diabetes are reduced. They exhibit pro-inflammatory signaling pathways, even if it doesn't turn into increased cytokine production. And it seems that they display an elevated recirculation to the bloodstream. And that's all. Let me just thanks all my collaborators that make this work possible. Okay, I think it's my presentation has been closed. So we are open to the discussion if there are any questions. Okay, I have a question. They're asking me if there are any differences between the pre-diabetic and diabetic patient. And I think that, just in the first slide that I showed you, we have pre-diabetic patients. So we evaluate the frequency of PD-1 positive cells in pre-diabetic patients, but we couldn't see any difference between them. And but for other studies, the RNA-seq and TCR-seq, we didn't check any difference between pre-diabetic because we chose only clear diabetic patient for RNA-seq and TCR-seq. So another question, do you have any idea if the PD-1 positive T cells are also present in the mice? Yes, there are some studies about the PD-1 T cells in the mice, and this seems they have a pathological effect in the visceral fat of mice. So, is there any other questions?
I don't want to go into depth of that. And then you can say, why is the incidence rate declining? That would be speculations. So yeah, let's just go to if there's anything to learn for such an observational study. Well, you can say the observation is that the incidence rate of hospital admissions with hypoglycemic episodes as primary diagnosis code declines. So that's at least one thing to learn. And you can say that new glucose lowering treatments might play a significant role in this. So clinicians could consider newer treatment alternatives to those treated with human insulins or sulfonylureas, if that is a possibility. But there are, of course, other considerations as well. Then these risk factors, in my mind, should be considered.
Models across all the performance measures. With that, I would like to acknowledge the beauty
combinatorial variance, which is very rare in real-world data. Any reason why negative binomial model is not used?
No, it was due to that was what was currently used in the literature that we saw. So we anticipated the Poisson regression as the tradition within that field of hypoglycemic events. But binomial models could have been used instead. I agree. And yes, the assumption might be violated in this study.
Great. And the next question that came in was that did you include exercise capability, as a lack of fitness is known as a major risk factor? That's an interesting question.
Let me just see it written. No, it would of course have been nice if we have had that a measurement of that. But no, we didn't have. We were not available or able to include these data. They're not part of the registries. But I can say that we are working on a diabetes database in Region North Denmark, where we will have a lot more data. Not much on exercise yet, though, but I do believe that it will come.
Great. And I guess following that question, do you have a hypothesis of what might be seen if you were to do that study looking at exercise capability?
Yeah, well, you can say that it probably would increase the risk a lot. But again, I think it's difficult to measure also. So with an exercise test, I would actually be interested to hear what could be done there for a precise estimate of that.
Yeah, for sure. I actually had a question as well with your analysis. Were you able to run any sensitivity analysis with regards to sex differences on these outcomes?
Oh yeah, I think I actually did that, but I'm not that. I can't remember the results that much, but you can say it was clear that that being female was associated with a reduction in risk of these hypoglycemic episodes leading to hospitalization. But I don't have. I won't go into details. What could be the reason behind that?
Fair. No, that was a great presentation, and I think a very important one. So thank you very much. We can move on to our next speaker. Our next speaker is Dr. Joseph O'Reilly from the University of Edinburgh. His talk is titled The Association of Hypoglycemia Exposure with Subsequent Adverse Events and Severe Hypoglycemia, Preliminary Results from HypoResolve.
Good afternoon. My name is Joseph O'Reilly and I'm based at the University of Edinburgh as a member of Professor Helen Clune's research group in the Institute of Genetics and Cancer. Today I'll be presenting some preliminary results from one part of the wider HypoResolve project. This particular piece of work is a quantitative assessment of adverse outcomes that have occurred as a consequence of hypoglycemia experienced during a clinical trial. Today's presentation is structured as follows. I'll first present an overview of the HypoResolve project and the motivation for this piece of work, and I'll discuss the qualities of the dataset that we've analysed and our statistical framework. Finally, I'll present our results, their implications, and how we intend to improve these analyses going forward. The HypoResolve project is an innovative medicines initiative funded project, which has the aim of reducing the burden and consequences of hypoglycemia for people with diabetes. HypoResolve's diverse work programme is spread across multiple work packages, and the work presented today forms part of Work Package 4. And this has been established to leverage large quantities of clinical trial data to investigate both predictors of hypoglycemia and the clinical consequences of hypoglycemia. Hypoglycemia has a substantial effect on the lives of people with diabetes. The explicit goals of our work package are to quantify the effects of known consequences of hypoglycemia and to also explore the association that hypoglycemia has with as yet unidentified consequences. The short-term effects of hypoglycemia are well known to people with diabetes and to diabetes healthcare specialists. But identification and quantification of medium and longer-term consequences requires the application of large data sets and statistical inference methods. To perform such analyses, we've utilized a pooled, harmonized clinical trial data set consisting of data provided by multiple industry and charity partners. This data set is of an unprecedented size and depth, with low-level, granular information available for each trial participant. Raw clinical trial data is delivered by pharmaceutical and charity partners to our colleagues at the Swiss Institute of Bioinformatics for pooling and harmonization. This process left us with available data from over 60,000 individuals across all trials. However, differences in the format and content of the recorded data across trials resulted in missing data for key covariates as a consequence of the harmonization process. We therefore focus our analysis on individuals with available basic covariates, discarding data from individuals with no diabetes type data or who came from trials with adverse event data that could not be placed into a longitudinal chronology. This process resulted in a large cohort of 22,000 individuals who had experienced almost 900,000 hypoglycemic events during their follow-up period. Our methods to investigate consequences of hypoglycemia are split into two main approaches to analysis. First, we investigate the relationship between prior and subsequent hypoglycemia events, and for these analyses we utilize a logistic regression framework, adjusting for sex, age, and diabetes duration. The exposure in these models is the count of hypoglycemia events experienced by an individual in the previous 45-day period, with the outcome being a binary measure of ever experiencing a hypoglycemia event in the subsequent 45-day period. This 45-day period was chosen as a medium-term horizon that maintains computational tractability. Second, we perform time-to-events analysis, and we use time-updated hypoglycemia exposure as a covariate in these models, alongside adjustment covariates of sex, age, HbA1c, and diabetes duration. We explore the association of hypoglycemia exposure with subsequent CVD, nephropathy, neuropathy, or severe hypoglycemia, each as an outcome in a separate model. We also make sure to analyze all data from people with type 1 and type 2 diabetes separately. A benefit of access to low-level information regarding hypoglycemia events is that we can define hypoglycemia arbitrarily, and this is regardless of how it was defined in the original clinical trial. Across all analyses, we consider 4 different definitions, and these are Level 1 hypoglycemia, which is an event with an associated blood glucose measure between 3.9 and 3.0 millimoles per litre. Level 2 hypoglycemia, which has an associated blood glucose measurement below 3.0 millimoles per litre. And Level 3, or severe hypoglycemia, in which the individual cannot self-treat the event, irrespective of the associated blood glucose level. As a further definition of hypoglycemia, we also investigated all recorded hypoglycemic events, irrespective of their associated blood glucose measure or their self-treatment status. Before presenting results from our regression analyses, we'll briefly look at the cohort characteristics. This slide presents a very brief overview of the incidence of hypoglycemic events in our analysed cohorts. Unsurprisingly, we observe fewer severe events than level 1 and level 2 events, and there's a higher incidence of hypoglycemia in people with type 1 diabetes than for people with type 2 diabetes. And here we have the general characteristics of a cohort at baseline. The cohorts for type 1 and type 2 diabetes are broadly similar, with people with type 2 diabetes, on average, having more complications at baseline, a slightly higher HbA1c, and they're slightly older at baseline as well. Now moving on to the regression results, we first present results for logistic regressions investigating the association between exposure to hypoglycemia and subsequent events, and we start with type 1 diabetes. So these plots show the estimated odds ratios for different exposures and outcomes, with each panel representing a different hypoglycemia outcome, and each row represents a different exposure. The colour of the point and range represents whether the estimated odds ratio and its 95% confidence interval is exclusively greater than 1. So what we see is that a greater number of any type of hypoglycemic event is associated with higher odds of subsequent events in the following 45 days, and this is shown by all the odds ratios being greater than 1. The associated confidence intervals for these estimates are particularly narrow, to the point of being difficult to see on these plots, and this precision demonstrates the benefit of analysing such a large dataset. The results for type 2 diabetes are very similar to those for type 1 diabetes in terms of the direction of the associations, and there's some small differences in the magnitude of these associations. We now present results from the time to event regression analysis, again starting with type 1 diabetes. As with previous slides, each panel here represents a different clinical outcome, with each row representing a different hypoglycemia exposure. Hazard ratio confidence intervals encompassing a value of 1 are presented in dark red, and hazard ratios with a confidence interval exclusively above 1 are in light red again. In further support of earlier results, in the second panel, we see a clear association between experience of hypoglycemia and elevated hazard for subsequent severe hypoglycemia events. One difference here, though, is that these models use a binary hypoglycemia exposure, and consequently these results show that experiencing just one hypoglycemia event is associated with an increased risk of subsequent adverse outcomes. We also see potential associations between level 3 events and increased cardiovascular disease hazard on the top row in the leftmost panel here. However, for this association, the confidence interval encompasses a value of 1. The confidence intervals seen here are slightly larger than those in the previous slides, and hopefully more data will improve the estimate precisions here. For type 2 diabetes, the results are again similar to type 1 diabetes, with the main difference being that for type 2 diabetes, we observe support for associations between level 2 or level 3 hypoglycemic events and subsequent elevated hazard for cardiovascular disease outcomes. This is presented here in the first panel on the left. There's also evidence that exposure to any type of hypoglycemic event is associated with elevated hazard for subsequent neuropathy. This is presented here in the final panel. Our results provide quantitative evidence that hypoglycemia is associated with negative clinical outcomes. It's important to note that exposure to events of any severity are associated with subsequent severe hypoglycemia. Severe hypoglycemia is known to have a heavy burden and to be associated with several particularly negative clinical outcomes. These results suggest that exposure to any hypoglycemia could therefore elevate the risk of a cascade of negative consequences. Associations between hypoglycemia and subsequent CVD are similar to those estimated in other published studies. However, our definition of CVD was based on the MEDRA system organ class, and this encompasses many milder CVD events alongside major adverse cardiovascular events. When considering the strengths and limits of our work, compared to the published literature, our cohort is relatively large, and although there have been large-scale national-level observational studies of hypoglycemia and its consequences, our cohort is unique in its scale and granularity of hypoglycemia event data, and this has allowed exploration of several definitions of hypoglycemia alongside one another. Conversely, our study was limited by the relatively short average follow-up time, which was between 1 and 2 years. This may impact our ability to accurately identify associations with longer-term consequences of hypoglycemia. The analyses performed here are also naturally conditioned on trial inclusion and exclusion criteria, and are therefore unlikely to be representative of a general population of people with diabetes. This is particularly important when considering conditions such as hypoglycemia unawareness, which often lead to exclusion from insulin trials.
About
in mouse islets, we treated PERK inhibitor, and we observed that AC3 conversion. It suggests that sorry.
No problem.
Sorry.
So don't worry. If you want to think it, you can write it in the question and answer dialog box, okay? And you can answer this question without worries, okay?
Yes.
We did a linear regression and adjusted for age, BMI, maternal weight gain, and
Therefore, the nature of this lesion per se, it was not exactly clear-cut, but that's the best definition that would, the best naming that people could come up with.
So do you think, so Ellie Pantolo has asked, is the dysmyelination process reversible? And how can the knowledge that you've gained in terms of up and down regulation molecules be used clinically? And can any of these
difficult to be diagnosed. It's a combination of the presence of the signs and symptoms of heart failure, plus the changes in left ventricular diastolic function. And in HFpEF, what we know that we have is the
Good evening, everyone. I'm really grateful to the Scientific Committee of the EASD for giving me the great opportunity to present this work, which is part of an ongoing research project where we are trying to define the critical pathological role of lipids in the etiology of diabetes. We all know that the early phases of the natural history of type 2 diabetes are characterized by insulin resistance and chronic hyperinsulinemia, which have additive detrimental effects on the progression of the disease. Now, while the pathogenetic role of insulin resistance is well established, the negative effects of hyperinsulinemia and its two main determinants, insulin hypersecretion by pancreatic beta cells and low insulin clearance, mostly by the liver, have been increasingly recognized over the past decade. Many mechanisms have been proposed to explain the development of insulin resistance and chronic hyperinsulinemia in the absence of substantial changes in plasma glucose levels, and among them, excess lipid availability is one of the most likely. In fact, chronic elevation in plasma triglyceride levels, the most common lipids in the body, has been consistently associated with an increased risk of type 2 diabetes in both cross-sectional and prospective studies. Furthermore, triglycerides have been associated with insulin resistance and with enhanced insulin secretion in previous human studies, regardless of potential confounders. So based on the available evidence, we hypothesized that hypertriglyceridemia could lead to insulin resistance, insulin hypersecretion, and possibly to reduced insulin clearance, altogether leading to glucose intolerance. These effects remain to be confirmed in experimental studies. and importantly, it remained to be established whether the actions of triglycerides are direct or mediated by their lipid metabolites, the non-esterified fatty acids or NEFAs. The fourth aim of the study was to compare the glucometabolic effects of mild acute hypertriglyceridemia alone or combined with an elevation of circulating NEFAs in non-diabetic subjects. To this aim, we analyzed available data from 2 clinical studies performed at our lab, which were prospectively designed to allow this comparison. In the studies, 22 volunteers underwent 2 5-hour intravenous infusions of either normal saline or intralipid, without or with heparin. The latter was used to activate the lipoprotein lipase and enhance the release of NEFAs. Oral glucose tolerance tests were then performed during the last 3 hours of the infusion. Insulin sensitivity was assessed after fasting by the OMA-IR and during the OGTT by the Matsuda index. Insulin secretion rate was estimated from C-peptide deconvolution, and insulin clearance was measured as the ratio between insulin secretion and plasma insulin. Beta cell function parameters were analyzed by mathematical modeling of glucose, insulin, and C-peptide data during the OGTT according to Maris model. The main clinical and metabolic features of the participants who underwent the lipid infusion without or with heparin are shown in this table. They were all healthy, lean, young individuals, and there were no significant group differences. During both lipid infusions, plasma triglycerides reached similar levels of around 2.5 mmol per liter, which is a high but still physiological level in the general population. On the other end, as for study design, NEFAs were markedly higher when heparin was co-infused with intralipid, as indicated here by the continuous red line. At baseline, that is, after 2 hours of lipid infusion, plasma glucose levels were unchanged, while plasma insulin was increased. And the latter effect tended to be more evident after the combined intralipid plus heparin infusion compared with the intralipid infusion alone, although group differences did not reach the statistical significance. Lipid-induced hyperinsulinemia, in spite of similar glucose levels, explains the worsening of the OMA-IR and was mostly driven by a reduction in insulin clearance in both groups, with only minor effects on insulin secretion, which again tended to be more evident after the combined intralipid plus heparin infusion. After glucose
so supporting the hypothesis that this event is mediated by splicing mechanism.
Age on any of these cardiovascular outcomes that were examined. That is, there was no difference in outcomes regardless of the age.
Peripheral nociceptor phenotype is characterized by deafferentation. And all of these patients underwent T1-weighted anatomical scan.
Slide showing that in people
this patient may have a higher risk of
A1C, weight, systolic blood pressure, and
academic degree, insulin therapy, and the degree.
We still don't know all the molecular mechanisms inducing the
So how do I say it? The baseline insulin.
So we use basically for all experiment 300 islets.
their waist.
Insignificant, but that was definitely a
40 academic
able to end.
Or below 75 with circles. The main point from this slide is that age did not impact on the beneficial effects of erdagliflozin to slow the decline in EGFR over time. This was true in the strata above and below the age of 65, as well as the strata above and below the age of 75, so that the beneficial effects of erdagliflozin on EGFR were sustained. This slide looks at the change in urine albumin to creatinine ratio from baseline over time. As you've already heard, the SGLT2 inhibitors are associated with a beneficial effect on albuminuria. This was seen in the erdagliflozin VERTIS-CV study, where there was a decline in the albumin to creatinine ratio initially with treatment, and then the albumin to creatinine ratio remained lower than the placebo-treated patients with erdagliflozin in both the strata above and below the age of 65 and the strata 75. And there was
say that type 2 diabetes increases in morbidity and mortality in heart failure, irrespective to the initial level of ejection fraction, that early detection, diagnostics and adequate, of course, treatment should be focused both by cardiologists and us, that the detection of HFpEF also goes into the whole spectrum of the impairments that we should evaluate, and the attempts to make a sort of integrated biochemical clinical parameters into risk score engines. In some of them gave promising results, signifying that it might be one of the approaches that we should follow. Thank you very much.
Yeah, Nebojša, thank you very much for this wonderful overview on the role of the diabetologist in heart failure and diabetes, and also highlighting screening and early detection. If I may start with a question before Doina, and we also have received questions from the audience. The terminology diabetic cardiomyopathy, is that still used? Is that still helpful to have this as a separate entity being classified? Or, I mean, we are more moving with the cardiologist's definition of heart failures and the different categories. What's your view?
Yes, I mean, from the very beginning, there were some proponents and opponents of the idea that diabetic cardiomyopathy exists. And it was very difficult to prove that you have some initial changes that are not related to ischemic changes, hypertension changes, and all others. But still, cardiologists have left more or less this or trying to say that this is one stage of heart failure. I still cannot overlook some data saying that there are some changes in the heart muscle that are underlying this state. Maybe they are the very initial changes underlying heart failure, but looking for diabetic cardiomyopathy might be important, and that's why it is still, in my opinion, should stay somewhere around. In this slide, I just wanted historically to show how the things went on. I'm aware of the fact that this is not universally accepted.
Thank you. Dorina?
Yes, we have a question here. Should we determine NT-proBNP, or should we just give all people with type 2 diabetes an CGLT2 inhibitor? What do you think, Nebojsa?
Although there are some situations in which they suggested that you should give one drug to all, like all statins to all diabetic patients and so on. I still believe that we should go for NT-proBNP, at least in the context of this algorithm from Toronto, and see whether we do have some changes. Although I know that NT-proBNP is not very specific, but we don't have anything better. So, and we have this fascinating data with SGLT2 inhibitors, but I think that we should have some more stratification how these agents do. Maybe somebody would show that, maybe Baruch would show that, but stratification how that works in non-diabetic patients, because the results that we have now exceeding diabetes are usually in a mixed cohort. So we still are not quite aware of the mechanisms through which GLP-2s are working.
So maybe in patients with long evolution of diabetes, we have to think of CGLT2 because, yes, the risk is very high. I think that Paul has a question for you.
Yes, thank you, Nebojsa. Very nice presentation. I'd like to go back to the assessment of the risk for heart failure. And you showed, for instance, the algorithm from Toronto group. And I would say, based on this algorithm, all the patients are at high risk for heart failure. It's not so much discriminant, I would say. But you mentioned, of course, albuminuria as a marker, risk marker, and also microangiopathy. Maybe we could consider all microangiopathic complications. There are some data, I mean, the Tayside study, for instance, and other data suggesting that the number of microangiopathic complications is associated with a higher and higher risk of heart failure. So it could be on the side of the diabetologist to say, well, these patients with several microangiopathic complications should be more assessed for heart failure. What do you think?
Yes, I think that we have some micro, like you said, for which there were studies and there were assessments, and then we can easily go with that. I would, for example, nephropathy or some others, I would be
by the ATC system. This first table is just to show you the matching characteristics. The mean age was 67 years, with 55% male subjects in the cohort. And now to population characteristics. As present before index date, we identified these characteristics by admission diagnosis and drug prescriptions, as we did not have access to laboratory results or questionnaires. In general, subjects with type 2 diabetes were unhealthier, more frequent smokers, alcohol abusers, and obese. In addition, we identified characteristics that could affect diabetes status and bone quality. For example, prior pancreatitis, thyroid disease, and glucocorticoid use. Lastly, we identified comorbidity using the Charlson Comorbidity Index that includes a large number of diseases in categories and provide a weighted index to predict morbidity. And as shown, subjects with type 2 diabetes had a higher index score compared to control subjects. Subjects with type 2 diabetes had a lower income and were more likely to be alone compared to control subjects. These characteristics are known to affect morbidity and risk of type 2 diabetes as well. This figure illustrates the odds of developing type 2 diabetes among those who have used alendronate. The first one ever use of alendronate was compared to never users, and the odds of developing type 2 diabetes were approximately 30% decreased among prior users of alendronate. Effective use refers to time of effective alendronate use and was compared to those with short duration of alendronate use as below 6 months as a reference. The odds of type 2 diabetes decreased with higher effective use. The bottom illustrates the effects of compliant use, estimated by dividing the cumulative dose by the treatment duration. The higher medication possession ratio, or MPR, the more compliant is the person. Though not significant, it seems that higher compliance could be related to decreased odds of type 2 diabetes. So I will try to make a cautious conclusion on these register-based results. These data suggest that alendronate may have the possibility to prevent or protect against later development of type 2 diabetes in a dose-dependent manner. However, these register-based studies are far from the truth, and we need to investigate the possible mechanism of action by alendronate on glucose metabolism in future clinical studies. So we hope to, and encourage to, investigate if this mechanism is present by, for example, testing insulin sensitivity as well as bone indices and glycemic control in both healthy individuals, people with prediabetes, and people with diabetes. And with these words, thank you very much for your attention.
Thank you, Dr. Vides, for this wonderful presentation. We have one question that came in so far, and again, I invite the audience to write in their questions if you have any. So the question here is, did you assess physical activity as well? Could it be that the reduced bone turnover in diabetic patients is in fact primarily caused by lack of physical exercise, which is typical for these patients?
Yes, thank you for that question. We didn't, and we didn't because we didn't have access to physical activity or exercise in the registries. So we didn't have the possibility to include this factor along with BMI, because it could be interesting to see, as written, it could reduce bone turnover even more in these patients, but we didn't have access to the data, unfortunately.
Have you come across any literature that may have suggested a relationship with this medication and participants who are physically active versus not for these outcomes?
That alendronate should affect Or what is
have you come across, or whether the physical activity has been assessed for this particular outcome, and what you would hypothesize would be seen if you did have this information?
Yeah, well, the interesting part is that people with diabetes have a decreased bone turnover, and alendronate actually decreased this bone turnover even more. But it seems to work, even though, and it is right that exercise may have the possibility to reduce bone turnover as well. I don't know if there's any interaction between exercise and alendronate use on bone turnover, unfortunately.
I see. So we have another question that just came here. There has been interest in vitamin D and diabetes. Vitamin D and bisphosphonates are often co-prescribed. Was this accounted for or looked into?
Yeah, we hoped that we could include vitamin D in the study, but we weren't able to because vitamin D is provided in Denmark as over-the-counter medicine. And these drugs that we looked in are linked to this unique security number, and vitamin D use isn't linked to this number, so we couldn't access vitamin D.
I see.
And I guess we didn't have access to...
Oh, no, sorry, go on. Finish.
And we didn't have access to laboratory results as well, so we couldn't even see the vitamin D level.
I see. Actually, this was somewhat similar to a question that I had as to whether you had other information with medication use of these participants and whether that was taken into account when you were running the analysis.
So you have to say again?
If you were taking into account other medications that the participants or individuals in this data were on, and if that was included in the analysis.
We included the glucocorticoids in our adjusted analysis, and we made some sensitivity analysis on people using antihypertensive medicines and treatment for dyslipidemia, anti-hypercholesterolemia drugs, and it didn't seem to affect the results. I reckon that those who were not treated for hypertension actually did have even lower odds for type 2 diabetes compared to those treated with antihypertensive drugs.
Interesting. And I actually had a question. I don't see any other new ones coming in, but I know this analysis was mainly with the Danish population. However, have there been other studies in different ethnic groups or ethnicities showing similar or different results?
I'm not sure I understand. We used the Danish population, yeah, and then you said about groups?
Yeah, well, have there been other studies looking at different populations and whether those are consistent with your findings?
I think there is two previous register-based studies. One actually on the Danish population as well, and another on Taiwan population, I think. And they are similar results as these, yes.
That's interesting. Great. I don't see any other questions coming in, so I think we can wrap that up. Thank you, Dr. Vigers, for that wonderful presentation. Our final speaker is Dr. Hans Biegelbach from Medical University of Vienna, and their talk is on sex-specific differences in patients deceased after bariatric surgery, a retrospective registry analysis.
Dear ladies and gentlemen, thank you for the opportunity to present the retrospective registry analysis of patients in Austria with a history of bariatric surgery. My name is Hannes Biegelbach, and I am a resident at the Medical University of Vienna at the Department of Medicine 3 at the Division of Endocrinology and Metabolism. First of all, let me present a brief overview and the aim of this data analysis. Bariatric surgery is a frequently performed procedure to treat morbid obesity with increasing numbers worldwide. It was shown that it can reduce all-cause mortality compared to non-operated controls. However, the mortality remains still higher compared to the general population. According to the IFSO report 2018, most of bariatric surgery candidates were women. Moreover, sex-specific motivations to undergo bariatric surgery were found. In men, the medical condition is of major importance, whereas in women, the appearance is the driving factor. Thus, men tend to be older and have a worse overall health at the time of the bariatric surgery compared to women. Furthermore, some studies showed that men have a higher perioperative mortality. The aim of the study was to analyze sex-specific differences in deceased patients with a history of bariatric surgery. A retrospective registry analysis of medical health claims data in Austria was performed. It is important to know that the medical health insurance found in Austria covers almost the total population with a coverage of approximately 98%, so the data were as representative as possible. Medical health insurance provided different data sources. First, demographic data with dates of birth, operation and death. Second, MEL codes, which are codes for medical single procedures, for example, HF 220 for sleeve gastrectomy. Third, ATC codes for reimbursed drug prescriptions. Fourth, ACD codes for primary and secondary diagnosis along with discharge date of hospital, and the number of days hospitalized with the main diagnosis based on the ICD code. The inclusion criteria was a bariatric surgery between January 2010 and December 2018. The bariatric surgery was identified based on following MEL codes: HF 220 and 230 for sleeve gastrectomy, HF 240 and 250 for gastric bypass, HF 254 and 255 for biliopancreatic diversion, HF 260 and HF 270 for gastric banding. The individual observation period ranged from 1 year prior to the bariatric surgery until the April 2020. Overall, 19,901 patients with 107,806 postoperative years of observation were eligible for the analysis. In total, 74% of all patients were women. The main focus was on sex-specific differences of comorbidities in patients. and antihypertensive drugs were also more common in men compared to women. Let me show now the main results of this study. Cardiovascular diseases were the most common comorbidities in men and women, with no sex-specific differences. Moreover, psychiatric disorders were found in more than 50% in both men and women. However, there was no sex-specific difference. In patients deceased after bariatric surgery, the proportion of patients with diabetes and the proportion of patients with malignancies were significantly different between men and women. Men had significant more frequently diabetes, whereas malignant diseases were more often found in deceased women. This brings me to the conclusion of this analysis. In this analysis, we could show that short-term mortality and long-term mortality was higher in men compared to women. In deceased patients, malignant diseases were more common in women, whereas diabetes was more common in men. And men had a higher preoperative intake of cardiovascular drugs, whereas women had a higher intake of psychopharmacological drugs. I would like to thank you for your attention and feel free to ask your questions.
Great, thank you, Dr. Haynes-Spiegelbuch, for that wonderful presentation. We actually just had a question come in. I'm going to put that up there. The question is, what about smoking and alcohol? Were you able to look into these?
Thank you for this question. I think that are two very important factors affecting mortality, but as the data are from the health insurances and the diagnosis are based on the ICD code, we are not able to make an exactly proportion of people who smoked in this cohort because the smoking status is not evaluated in this data. But that would be a very important thing to analyze in further studies, of course.
And I think the second part was about alcohol, whether that was also available.
That's a very good point. Some patients were diagnosed with substance disorders, abuse, but we don't exactly know which type of substance were used. But that would be an important question to further analyze, yes, of course.
Definitely. Did you have information? Oh, I guess, did you want to finish up on that point?
And also the sex-specific differences on substance abuse is also a very important thing. And this might be higher in men, and therefore maybe is also in affecting the worse mortality rate in men after bariatric surgery.
For sure. My question was actually also along the lines of this, but did you have information on diet from these participants?
Unfortunately not, because we have three data sources. There are diagnoses made from hospital, the drug intake, and the MEL codes, but we don't have data on metabolic data information such as body mass index or dietary habits. But that would be a very important thing to strengthen the study, of course.
For sure, and I think sorry, I keep cutting
no problem. But that's maybe the limitation of such studies with medical health insurance data. That there is not every we have a huge amount of data, but but in one individual person we don't have the data of the all necessary data of the individual person. That's the problem of such studies and analysis.
Yeah, it's a very large cohort, but of course there are major limitations when they're using these registry data. And for sure, I think having some type of dietary information as well as physical activity and smoking would definitely strengthen what we're seeing here. My other question was with respect to your results for the different comorbidities that you focused on, what variables were you adjusting for those analyses, looking at the sex differences for those different outcomes, different comorbidities?
What do you exactly mean?
You showed the results for diabetes, yes, cardiovascular disease. Were you adjusting for different variables in that analysis as well? Or was that just looking at those comorbidities?
No, it was just let's say descriptive statistics, and we don't make corrections for let's say multiple testing or something like that, because it's more like a hypothesis generating study, because we have to plan based on this study. We have to plan a well-matched prospective cohort study to make profound predictions of mortality, I think. But that was not the aim of the study, we have to say. The aim was in this real-world registry analysis to have to look how the mortality is influenced by sex.
I see. And I don't see any other questions coming in, but my final question would be, where do you see this work moving forward? I think it's interesting to see the sex differences, but what would be the next steps here? Maybe the
most important step is, based on this study, we see that men had a worse mortality outcome, and we think that it's because of the worse overall health at the time at the operation, because men tend to be older at the operation, and maybe that's the biggest influence on the mortality outcome after bariatric surgery. And so I think it's important to focus on when on preoperative counseling, especially for men. We have to empower them to undergo earlier bariatric surgery, and maybe this would improve the mortality outcome.
For sure, I think a very important aspect to focus on in the future. I don't see any questions coming in, so I think that wraps up the discussion session for your talk. Thank you very much. And this ends the oral session for today as well. Thank you. I would like to thank the speakers for their excellent work and presentations, as well as I thank the audience for their engaging questions and making this session very interesting. Please reach out to the speakers if we were unable to get to your questions. I know some were coming in after the talks had already been done. And with that, have a wonderful rest of the day and enjoy the rest of the conference. Thank you, everyone.
With users and potential users of the e-learning platform. All of this work has helped us to achieve one of the platform's principal goals for 2021. It was clear from user feedback over the last couple of years that learners really want to be able to earn continuous professional development (CPD) points from doing our courses. This summer we submitted our first module for CPD accreditation by the UK's Royal College of General Practitioners. We're delighted to announce now that our submission was a success, and so far one of our modules has been given CPD accreditation, with another 6 in the process of receiving accreditation. The entire site is also being submitted for accreditation by the Royal College of Physicians in October, and plans are underway to submit our online courses to national accrediting bodies around the world. So why not create your free account today? Join us and a host of well-known faces from the world of diabetes research, treatment and management, and develop your professional learning alongside the experts.
risks and clusters of infection that may occur in that setting. Also, the impact of social distancing and social isolation on the individual, specifically mental health and the impact on their diabetes with changes to mental health. Not just that, the ability to obtain medication and administer the medication.
into an established resource of systematically reviewed, clinically focused education for the whole diabetes care team. We now offer more than 70 modules, each one carefully crafted with and presented by leading experts from around the world, with course topics ranging from therapeutic inertia and phenotypic variability to metabolic surgery and islet cell transplantation. The site is visited by an average of 2,300 visitors every month and has an audience that is truly international, hailing from every inhabited continent on the globe. We also reach a broader healthcare world via social media and are building an enthusiastic community of followers, currently standing at over 2,700 on Twitter. This year saw the launch of a new section on the EASD e-learning platform, Horizons. In this section, we provide visitors with weekly editorial updates on diabetes news from conferences and journals, alongside our range of filmed initiatives. Horizons is now home to over 100 educational films, looking at the latest innovations and debates in diabetes research and treatment. Last year, our series Around the Diabetes World in 80 Days brought together specialists from around the world to highlight the particular challenges of diabetes care in their region. And with the ongoing COVID-19 video series, we were able to respond to a fast-changing information landscape, sharing vital information on diabetes and the pandemic as and when it arose. 2021 being insulin's centenary year, we have marked the occasion with a series of 100 insulin-related features, ranging from the history of insulin to the insulin replacement therapies of the future. Also new this year is the long and the short of it, in which eminent experts interview one another on a key aspect of diabetes treatment or research. Finally, there is the patient who changed the way I think about diabetes, our podcast series in which leading diabetes clinicians describe the cases that have shaped their vision and understanding of diabetes care. We have plenty more exciting new initiatives ready for release, so do keep a lookout on Horizons over the coming months for, amongst others, the briefing room, 5 things we don't yet know, and insulin in my country. Meanwhile, of course, our core roster of courses keeps growing month on month. Just this week at the EASD's second virtual annual meeting, we will be launching 5 new modules covering the treatment of NAFLD, painful diabetic neuropathy and fear of hypoglycemia, plus new courses on obesity and diabetes and on real-world evidence. Further courses, including beta cell physiology, lifestyle interventions, structured education, and of course insulin, are in development for 2022. Over the last year, with support from our authors, expert reviewers, assessment setters, and several of the EASD's study groups, we have worked hard to improve the assessment aspect of our modules and increase their focus on practical clinical scenarios. 40 academic reviewers are now set up on the site to support its content development. And we have a new messaging and feedback system launched in May to help us communicate better with users and potential users of the e-learning platform. All of this work has helped us to achieve one of the platform's principal goals for 2021. It was clear from user feedback over the last couple of years that learners really want to be able to earn continuous professional development, CPD points, from doing our courses. This summer we submitted our first module for CPD accreditation by the UK's Royal College of General Practitioners. We're delighted to announce now that our submission was a success, and so far one of our modules has been given CPD accreditation, with another 6 in the process of receiving accreditation. The entire site is also being submitted for accreditation by the Royal College of Physicians in October, and plans are underway to submit our online courses to national accrediting bodies around the world. So why not create your free account today? Join us and a host of well-known faces from the world of diabetes research, treatment and management, and develop your professional learning alongside the experts.
someday when my ship comes in, she'll understand what kind of guy I've been. And then I win. And when she's walking, she's loving, so fine. And when she's talking, she'll say that she's mine. She'll say I'm not so tough just because I'm in love with an uptown girl. You know, you see her in the white bread world, as long with anyone with a blood can. Now she's looking for a downtown man. That's what I hear, yeah.
Hello, everybody. Welcome to the Albert Reynaud Prize Lecture. I am Chantal Mathieu. I'm an endocrinologist from Leuven, Belgium, and I am Senior Vice President of the EASD and Chair of the Albert Reynaud Prize Committee. The Albert Reynaud EASD Prize is awarded for outstanding achievements in research on the islets of Langerhans, outstanding contributions in histopathology, pathogenesis, prevention and treatment of the complications of diabetes. Of course, you need to be a member of the EASD to be able to apply to the Albert Reynaud Prize. So a message for these young and not so young researchers out there who believe that their work in this domain is outstanding. Do join us and have your friends nominate you for the Albert Reynaud Prize. This year's winner is our good colleague and friend, Pedro Herrera, from the University of Geneva in Switzerland. And throughout his career, he has made groundbreaking discoveries relating to pancreas development, and more recently also in the area of regenerative medicine and cellular plasticity. Professor Herrera described the versatility of pancreatic islets, first of all in adult mice, and then brought first evidence that the adult pancreas has the capacity of reconstituting new insulin-producing cells, thus having the potential to lead to diabetes recovery. Such mechanisms involve a high degree of changes in cellular identity, and in particular, Pedro has really cornered this interconversion of different cell types within the pancreas, and that was a phenomenon where he was a pioneer in describing it. And also, he found that this phenomenon also happened in human pancreatic cells. I know Pedro as a master in tool discovery, always happy to share his knowledge, to share his tools in the world of diabetes, and he's also introduced lineage tracing in the pancreas development field. So let us now listen to Pedro's lecture entitled Diabetes Remission Through Insulin Secretion by Islet Non-Beta Cells. Pedro, congratulations on your Albert Reynaud Prize.
Thank you very much. Thank you, Chantal. Chairwoman, dear colleagues, ladies and gentlemen, it is for me an honor to receive this 15th Albert Reynaud Prize, and a joy. And very precisely, 100 years after the discovery of insulin. As you know, insulin was the first peptidic hormone to be discovered, and 30 years later was the first hormone. The first protein that was fully sequenced, indeed, by Sanger. And later, it was also the first protein to be produced by DNA recombinant technology. And this joy for this award for me is even more intense since Albert Reynaud was working here in Geneva. And he was the mentor of two of my mentors. One was Jean-Dominique Vasali, an expert in messenger RNA stability. And Albert Reynaud was also mentoring earlier the young Lelio Orci, who you must know, no doubt. Albert Reynaud was also a close friend of, a close colleague also of my third mentor, who was Pierre Vasali, who at the time was the director of the Department of Pathology. So these three persons marked me forever, professionally, but also at a personal level. Anyway, the measure of success in our careers, in any professional endeavor, relies on the continuity and sustained progression of work. Today, I'm very pleased to show you a selection, a superficial overview of what are, in my view, my main contributions to science. Basically, what I'm trying to do in my laboratory is to understand the origin of insulin-producing cells, whether during normal embryonic development or during regeneration in diabetes. Therefore, my talk today will have two parts. First part will be dedicated to the developmental studies, the early studies, and then the second part will be more focused on the regeneration studies. What I do in the lab basically requires two experimental approaches. On one hand, we have the use of primary human islets, human islet cells, and on the other hand, we use transgenic mice. We use these mice so that we can, with the Krylov-P system, activate genes, in particular reporter genes, more specifically in an inducible way, to achieve what we call the tracing of cell lineages in the body. We also use the Krylov-P system to inducibly inactivate genes, the knockout mice, genes that are important for development or regeneration. And we also use mice with diphtheria toxin to inducibly and in a selective way ablate specific types of cells in the body. So why study embryo development? Why the development of the pancreas? I used to tell my students that because by understanding the construction of the body, the construction of the pancreas, in this case, the construction of the organ, we will understand better the pathological basis of disease. So the first question I wanted to address when I was becoming an independent scientist was concerning a dominant view at that time, namely that islet cell types, in particular beta cells, derive from embryonic glucagon-expressing alpha cells. And to address that, what I decided to do is to use the newly developed technique of Krylov in transgenic mice that was devised normally to inactivate genes, to make cell type specific knockouts. What I was trying to do in my early laboratory was precisely the opposite. I was trying to activate a gene that I was calling the reporter transgene to irreversibly mark cells in the body. So this way I would be able to trace back these cells to a developmental origin, and this is the basis of the in vivo cell tracing studies in mice. And this was the first use of this technique, the Krylov-P, to label cells in this way. Basically, the system consists of having doubly transgenic mice, whereby they have one transgene, which I call Tagger transgene, in which a cell type specific promoter in this first experiment, this was the glucagon promoter, insulin promoter, and basically PP and PDX promoters to drive the expression of Cre recombinase in these specific alpha, beta, and so on cell types. And the second transgene is an inactive transgene that I call reporter transgene, in which there is a stop codon, transcription termination cassette that is flanked by two LOXPs and is in front of the coding region of a reporter gene. In this example, is beta-galactosidase. So in doubly transgenic mice, in the cells in which there is expression of the genes of interest, there is Cre activity, and this activates the expression of the reporter gene. And this is irreversible. And the progeny of these cells, if these cells are progenitors to other differentiated cell types in the body, are labeled forever irreversibly. And this way, we can trace the origin of these cells to a developmental origin. In this initial study, I could find that the glucagon tagger is not expressed in the beta cell lineage, and the insulin tagger is not expressed in the alpha cell lineage, meaning that these two alpha and beta cell lineages arise independently during ontogeny from a common precursor expressing PDX1. I was also able to show that the two cell types were derived from progenitors expressing PDX1. Then we wanted, in these early days, to analyze as many markers as possible with this new tool to try to define the profile of different pancreatic precursor cells. And then I generated a number of strains of mice using these promoters and other promoters, and also using mice generated by colleagues all over the world. And we were crossing these tagger lines with the newly developed reporter, universal reporter mouse that was called R26R, the ROSA mice. Basically, with these early experiments, we could find a number of things, like confirm, for instance, that PD-L1 is expressed in the progenitor cells that will give rise to all cell types of the pancreas. We confirmed that neurogenin 3 expressing cells are islet progenitor cells. And we can also find that nestin expressing cells are expressed in the progenitors of the exocrine pancreas, of the exocrine lineage, but not in the progenitors of the islets, as was proposed at that time. Incidentally, using these nesting tree lines, 2 different lines, we could also find, because of the low penetrance of the target transgenes, that in fact acini have a polyclonal origin because of this mosaic expression pattern. So one single acinus has an origin from from several progenitor cells. At this time, I wanted. I mean, we were doing so many experiments during this period. Today, for the sake of time, I cannot show many of the things I would like to show, but during this period, we also made experiments aimed at trying to understand how organ size, the cell numbers, is established during development. We made mice with a selective pancreas-specific inactivation of a number of key genes involved in the coordinated expansion and differentiation of progenitor cells. And in particular, we were exploring the Wnt beta-catenin MYC signaling pathway, which is very important during development. And we found interesting phenotypes of pancreas hyperplasia and pancreas hypoplasia. And we also found, by the way, that the pancreas-specific inactivation of MYC leads to a degeneration of pancreatic acinar cells that transdifferentiate into white adipose cells. This also happens with aging. Like I said, today, for the sake of time, I would like to just show you one experiment that was designed to further explore in an innovative way, and indeed, the differentiation potential of these neurogenin 3 expressing progenitors. Like I told you, neurogenin 3 expressing cells are, at least at the population level, multipotent progenitors that give rise to all cell types that we can find in an island, endocrine cells. So the idea of doing this experiment came to me after a conversation in Paris with my friend Raphael Sharman. I decided to do this in the following way. We know that at the population level, neurogenin 3 cells are multipotent. The question that I was trying to address was whether at the single cell level, these cells are multipotent, very much like bone marrow stem cells, for instance, or on the contrary, whether at the single cell level, each of the neurogenin expressing cells are indeed unipotent, committed precursors. So to explore this very specific question, one needs to do an in vivo clonal analysis. This is not a conventional cell lineage tracing. Indeed, this is a system in which you want to label only very few, very rare cells in the body of the embryo. And for that, I used the newly developed at the time reporter line, which was called MADAM. MADAM is an acronym for Mosaic Analysis with Double Markers in Mice. So the idea is to label as few cells as possible in the embryo, in this case neurogenin 3 expressing cells, and to follow the progeny of these cells and to see if these the clusters that are formed from these labeled progenitors are heterogeneous, then we will have the proof that these cells are multipotent. But on the contrary, if we see clonal descendants of these early labeled cells to form homogeneous clusters, isotypic clusters containing only one type of cell within islets, then this would argue in favor of the unipotency possibility. So we use these MADAM mice in which there's no one reporter, but 2 reporters that are inactive. And they are located on the 2 homolog chromosomes. In one allele, we have the N terminus of the green fluorescent protein, followed by the C terminus of the red protein with the LOXP in between. On the homologous chromosome, we have the opposite, the N terminus of the red protein and the C terminus of the green protein. So in cells that are not proliferating in G1 or G0, if there is clear recombinase activity, we may have this chromosomal translocation, which is a very rare event, indeed. And this is why we can use it as a clonal analysis marker. And in this case, we activate the expression of the two reporters, and the cell becomes double labeled, double color. The cell becomes red and green. On the contrary, if the neurogenin 3 cells, the cells that we were to analyze, are proliferating, are in G2 phase after the synthesis of DNA, then what recombinase can do is a translocation between sister chromatids. And in this case, we have, again, the constitution of the two reporters, green and red, but depending on the segregation pattern of the chromatids during the M phase, during mitosis, then we may have two different outcomes. Either one daughter cell is green and the other is red, and the other possibility is that one of the daughter cells is not labeled at all and the other is red and green. So by analyzing the color pattern of the progeny of the cells, we will know whether the precursor was proliferating or not. And very precisely, we have the proportions of cells with the different colors. So what we found with this analysis is that the clones of cells, islet cells that were labeled in the adult pancreas, indeed were homogeneous most of the time. The majority of the clones were homogeneous and very small in size. So the cells had, in addition, the two colors. And interestingly, in newborns, these labeled cells were found isolated within the islets of these newborn mice. Interestingly, these clones contain, on average, 2 cells at the age of 2 months, and they contain, on average, 4 cells in very aged mice at the age of 10 months. So, in summary, what these experiments were very clearly suggesting is that each single neurogenin 3 expressing cell becomes one islet cell at birth. So these are non-proliferating post-mitotic cells that are unipotent. They are indeed late precursors. They are not like the marrow cells. They are not multipotent. And in addition, incidentally, these experiments were also telling us, they were confirming that islet cells have a very long lifespan and they have low proliferation rates. So now we will switch to the regeneration studies. The central question, everything we do in the lab, basically almost everything we do in the laboratory, rotates around this very question. Can the adult pancreas regenerate new insulin-producing cells after a total beta cell loss, like in type 1 diabetes? So when I started this project around 1999, I tried different transgenic approaches, and this one was the most successful. What I wanted to have is a mouse, a healthy adult mouse, in which I could remove all the beta cells. So I didn't want to have a model of diabetes, but an experimental model, simplified model of beta cell ablation in mice that are otherwise healthy and completely normal. So the most successful approach that I could develop was this one. I was using an insulin promoter to drive the expression of the human receptor for diphtheria toxin. Mice are insensitive to diphtheria toxin. So in these transgenics, the injection of diphtheria toxin leads to the destruction of the beta cells, massive destruction, and with this tool, we then could try to answer the question we were asking, which is whether if we keep these mice alive with insulin therapy, there is regeneration in an adult pancreas, because this is the question we wanted to address. In an ideal situation, can the adult pancreas reconstitute new insulin-producing cells when they are lost? And the answer was yes. I was very surprised because I was convinced that that was going to give a negative result. But indeed, we saw that, in fact, many insulin cells were popping up in the islets of these mice that were completely devoid of the beta cells. Then the obvious question was, what is the origin of these cells? And we started to combine the ablation of beta cells with the labeling of other cells within the pancreas. And in this very example that I'm showing you here, we have a conditional tool to label specifically adult alpha cells. So in these mice, because of these 4 alleles, when we give the tetracycline analog doxycycline to mice in drinking water for a week or 2. In these animals, in this example here, in alpha cells, which produce this transactivator, which is dependent upon doxycycline, this binding allows this transactivator to activate the expression of recombinase. It binds to the threat-responsive element, which is driving CRE. And in the presence of CRE, there is recombination of the reporter and then activation of the expression of the reporter protein, which is a yellow fluorescent protein in this example. So by giving doxycycline, we label adult alpha cells in an adult mouse that is fully normal and fully healthy. And then we can start the experiment, which is the ablation of the beta cells, because we have the beta cell suicide transgene here. By providing this toxin, we destroy the beta cells, and we have, in this example here, the alpha cells label. So if we keep the mice alive for regeneration, we can see what happens. So this is clearly a pulse and chase approach. You can see here, we pulse the alpha cells and we chase them later. And as expected, this labeling was reversible. In these animals, glucagon cells remain reporter positive all the time. And interestingly, with time, some of these cells also started to co-express insulin, as you see here in this example. And even more interesting than that, we also had cells that were producing insulin, that were not making glucagon, but they were reporter positive. They were expressing the YFP protein, indicating that these were reprogrammed adult alpha cells that started to make insulin. So with these kind of approaches, we started to find different things that can be summarized in these two main observations. What we saw is that juvenile, adult, age animals spontaneously regrow new insulin-producing cells when they are massively lost. And that was the first surprise, like I said. And then the second surprise was when we realized that in this situation, the new insulin-producing cells in adult mice are indeed reprogrammed non-beta cells, cells that are transiently bihormonal, and then they stop making the first hormone. Cells that make glucagon and insulin, somatostatin and insulin, or pancreatic polypeptide and insulin, and then they stop making the first hormone. And this was the second surprise, and this surprise was even bigger, as you can see here, by comparing the font size of these two surprises. Much bigger indeed. So very quickly, we figured out if we were able to push all alpha cells to make insulin, what would happen? What would be the consequences of a massive alpha cell mass reduction? And we did this mice in which we can destroy the majority of the alpha cells with a glucagon DTR transgene, and we could see very clearly that there is normal glucagon signaling with a very small fraction of the alpha cell mass remaining. And also the beta cell function was perfectly normal, insulin tolerance, body weight, and etc. So with very few alpha cells, the mice seem to do very well. Another question we wanted to address immediately after these initial observations was whether the reprogramming of non-beta cells required the near total loss of beta cells to happen, and whether the mode of beta cell death is important or is determining this plasticity. Whether beta cells die by apoptosis or by necrosis is important. We wanted to address that, and for that we started to use streptozotocin and alloxan. These are the most popular experimental tools to have diabetic mice, diabetic rodents. As you know, these two molecules are glucose analogs and enter into beta cells through the glucose transporter, GLUT2. Interestingly, both produce the death of beta cells, which is of necrotic style. This death by streptozotocin and alloxan is necrotic, and also with these drugs, normally scientists can achieve something between 70 to 90% of beta cell removal. On the contrary, the diphtheria toxin works very differently. The toxin, as is secreted by this Corynebacterium diphtheriae, has three subunits, A, B, and T. B is the ligand of the human receptor, which is this heparin-binding EGF molecule, and once bound, it is internalized, and within these endophagosomes, with the change of pH, the T subunit, which is the transmembrane domain, undergoes a conformational change, and this translocates the A subunit, which is the active part of the toxin, into the cytoplasm. And once into the cytoplasm, what the DTA does is to catalyze the adenoribosylation of elongation factor 2. And this results in a very impressive and efficient inhibition of protein synthesis and the death of the cell by a process of apoptosis. So what are the differences between streptozotocin and diphtheria toxin? On one hand, we have the number of cells that is killed. With the streptozotocin, you have a remaining beta cell mass of 10 to 30%. With the diphtheria toxin, you only have 1 to 2% of beta cells left. And also the second difference is that strep induces necrosis, and while DT induces apoptosis. So we did this experiment to analyze the plasticity of alpha cells and somatostatin delta cells by killing beta cells with alloxan or somatostatin or diphtheria toxin. And we saw that in all conditions, we always could detect alpha and delta cell conversion, as you can see here with the different tracers here and there. So alpha and delta cell reprogramming does not require near total beta cell loss and is not dependent of the mode of beta cell death, apoptosis or necrosis. We also wanted to know what is the nature of the signals that guide this alpha cell conversion. In particular, we wanted to know the influence of glycemia or the influence of the pancreatic environment on alpha cell plasticity. And for that, we did a number of islet transplantation experiments. We were putting donor islets, wild type or transgenic, with the DTR transgene, into host mice that were immunodeficient, that were also wild type or transgenic. So the idea was to induce the beta cell loss in the pancreas or in the graft or both, and see whether this plasticity of alpha cells is dependent upon the glycemia of the mouse or on the influence of signals from the exocrine pancreas. So when we put wild-type islets in a transgenic host, we destroy the beta cells in the pancreas, but the mouse remains normoglycemic. And inversely, we also can destroy the islets in the graft only in a mouse that is normoglycemic. And what we observe is that every time we destroyed beta cells, independently of whether the mice were normoglycemic or hyperglycemic, independently of whether the islets were the endogenous islets in the pancreas or the graft islets in the kidney, in these cases, always we saw the conversion of alpha cells, but only in the islets in which there was beta cell ablation. So these observations were very surprising also, and were telling us that this alpha cell reprogramming occurs independently of glycemia, independently of the total beta cell mass, independently of signals from the exocrine pancreas that would be acting locally around the islets, and also independently of a functional immune system. So in conclusion, this alpha cell reprogramming appeared to be islet autonomous. This was very surprising. We then decided to ask the question in the opposite direction. Since only a small fraction of alpha cells spontaneously reprogram to become insulin producers after beta cell loss, we wanted to know what are the signals that restrict this plasticity. And we had this idea that the local deprivation of insulin resulting from the beta cell loss could be responsible for this alpha cell conversion. We have this idea because collaborating with Shang Sang in Lyon, we have been able to inactivate this tumor suppressor gene, MEN1, both in beta cells and in alpha cells. And the inactivation of MEN1 in alpha cells lead to the appearance of glucagonoma. So in these animals, in few months, the alpha cells have formed this tumor, the glucagonoma, but surprisingly, with time, this glucagonoma transdifferentiated into an insulinoma. These menin deficient alpha cells are starting to become insulin expressors. They became bihormonal cells, as you can see here, and with the trace, you can follow them, these cells became insulin-producing cells only. So glucagonoma transdifferentiated into insulinoma, and we thought that perhaps this was because of the lack of insulin within this lesion. So we tried to explore this, and there's two ways to do that. Either by inactivating the insulin receptor in alpha cells specifically, or pharmacologically by blocking the insulin signaling. And here I'm showing you this example. we use this molecule S961 from Novo Nordisk, which is an insulin antagonist. So we were giving this drug to mice in which we had pre-labeled adult alpha cells with doxycycline. And in this situation, what we could see is that the conversion of alpha cells was even more efficient than when we ablate the beta cells, indicating that insulin signaling blockade induces insulin production in a fraction of alpha cells. And also telling us that cell death, beta cell death itself, is not required for insulin gene induction in alpha cells. If we stop insulin inhibition with S961 by withdrawing this drug and keeping the mice alive for an additional month, what we observed is that there was a restoration of glucagon production by this fraction of alpha cells that are engaged in insulin expression. So insulin induction in alpha cells appear to be reversible. With this kind of experiment and other experiments, we could identify a number of signals that function as intra-islet constitutive breaks that are maintaining islet alpha cell identity and cell identity in general, like insulin or smoothen and other signals. And by removing these breaks, by blocking these signaling pathways, we could see that alpha cell identity is perturbed because of the induction of antagonizing forces. So at the population level, some of the alpha cells acquire an enhanced alpha cell identity, while other alpha cells acquire beta cell specific traits. And in addition, this is a reversible process. So these experiments were suggesting that intra-islet insulin is a constitutive break restricting alpha cell plasticity. And more general, I think that these kind of experiments are clearly suggesting that the majority of the more than 200 differentiated cell types in the body maintain their identity through an active process which is mediated by repressive signals that are blocking an intrinsic trend of cells to change. And I think that this is an important concept. So this is also another experiment that I want to show you to explore the discriminating power of full beta cell ablation. We wanted to see how glucagon signaling is altered in situations of massive beta cell loss. So Roger Unger, at the end of his career, launched this very provocative hypothesis. So that hyperglycemia in diabetes is caused by glucagon excess, not insulin deficiency. So he was showing that because in glucagon receptor knockout mice, there is no diabetes induction when they are given streptozotocin. So he thought, and he said that glucagon signaling blockade is sufficient to recover from type 1 diabetes without insulin. So what we wanted to explore was whether this hyperglycemia is caused by glucagon excess and not by insulin deficiency. And what we did is a number of experiments to see if glucagon suppression prevents hyperglycemia in the diphtheria toxin model, in which the ablation of beta cells is much stronger. So we use glucagon receptor knockout mice and anti-glucagon receptor neutralizing antibodies, and we use the transgenic mice to ablate beta cells, or we also use streptozotocin and the insulin receptor antagonist. So when we gave these animals diphtheria toxin, they became diabetic slightly less rapidly than controls, but the glucagon receptor knockout mice also became diabetic, meaning that glucagon signaling inhibition per se is not preventing hyperglycemia. So again, we check for the difference between streptozotocin and diphtheria toxin, and also in the glucagon receptor knockout mice, we observe that diphtheria toxin is more efficient than streptozotocin, and that streptozotocin is not causing a complete insulin deficiency. We gave mice doses of streptozotocin, and then we treated in insulin antagonist, and we saw that insulin complete insulin inhibition, the strep dosing-treated mice also became hyperglycemic, meaning that normal glycemia after strep depends on residual insulin. We also did the opposite with a very high dose of strep. The normal high dose of 225 milligrams per kilo, treated them with anti antibody. And in this situation, only the mice that had the highest dose of the streptozotocin, while the animals with the normal high dose, 175 milligrams per kilo, became again normal glycemic with the inhibition of glucagon signaling, meaning that residual insulin signaling is essential for normalization of glycemia in these models. So these experiments were suggesting clearly that the lack of glucagon signaling is not sufficient to prevent hyperglycemia. Residual insulin action is necessary to maintain normal glycemia, and however, clearly glucagon signaling inhibition improves glycemia in beta cell ablated mice. Now, what about plasticity in human pancreas? We wanted to do these experiments to provide a proof of concept that the facultative induction of cell plasticity in the human islet non-beta cells is something that also is possible. So we know, we have observed in the pancreas of patients with diabetes type 1 or type 2 the presence of bihormonal cells, like you see here, somatostatin and insulin, or here these cells containing glucagon and insulin. Also, studies by Klaus Kessner ex vivo using histone methyltransferase inhibitors reveal that this inhibition results in the appearance of bihormonal glucagon and insulin producing cells. So what we did in the laboratory is to use human islets from non-diabetic or from type 2 donors, dissociate them, and obtain very pure populations of the different fractions of cells. Here, I'm showing you the alpha cell fraction. And what we were doing is to induce with adenoviruses the expression of a reporter gene, the green fluorescent protein, and inducing the expression of different transcription factors. And after that, these alpha cells are reaggregated, and we form what we call monotypic alpha cell only pseudo-islets. This is one example of such a pseudo-islet in which you can see the human alpha cells fluorescently labeled. So what we did with these experiments is a very powerful approach. You can imagine clearly that we can do a number of in vitro analyses, we can do transplantation analysis, and in addition, we can play with the composition of these pseudo-islets. We can have pseudo-islets of beta cells only, or alpha cells only, etc., and with different proportions. We can also play with the number of cells that we put within each of these single pseudo-islets. So we have here a powerful tool to study human islet cell biology and label human primary cells very much like we do in mice with this pulse and cheese approach. We can lineage trace these human cells in long-term studies. So here, this is a pseudo-islet of human alpha cells that were transduced with PDX1 or with MAFA or with the two factors. And you see that after a week, about 40% of the alpha cells start expressing insulin. And more interesting than that, these cells secrete insulin in response to high glucose. And this is true even while at the beginning these cells are still expressing glucagon and the transcription factor ARX, which is expressed in normal alpha cells. So human glucagon expressing alpha cells can start also, like in mice, the production of insulin and the secretion of insulin in response to glucose in the appropriate conditions. So we wanted to know then whether these human alpha cells that make insulin are functional in vivo, and then we did a number of transplantation experiments. So I'm showing you here the very first experiment. We were able to ablate the beta cells in 6 mice. 3 of them were left untreated, and 2 of them received each 4,000 human islet equivalents, and the mice became normal glycemic, as you can see here. In this early experiment, we could also give the same amount of human alpha cell pseudo-islets making insulin from 3 donors, and we could transfer them into 1 mouse, which became as well normal glycemic. These recovered animals had all of them a normal glucose tolerance. We could detect in all of them circulating human insulin before and after the glucose challenge. Interestingly, when we removed the graft, the animals became diabetic again, indicating that insulin-secreting human alpha cells can lead to diabetes remission in mice. So these are the human islets engrafted under the kidney capsule after a month. You see here the beta cells in red and the glucagon cells in blue. And here you have several pseudo islets of human alpha cells that were transduced. You see the reporter gene, the green molecule here, and you see that nearly all of these cells are making insulin now. Indeed, after a month in vivo, the majority stopped making glucagon. So for some reason, which we don't know yet, this in vivo environment promotes the reprogramming and maturation of human alpha cells that make insulin. So you see here that human alpha cells can lineage trace in vivo, and they are indeed very stable. In the longest experiment we did, up to 6 months, we could see that these human alpha cells were still labeled, were lineage trace, and they were still making insulin very happily. So these experiments were teaching us that glucagon-expressing human alpha cells are stable and can engage insulin production and glucose-dependent insulin secretion and lead to diabetes remission in mice. And we also found the same in alpha cells from diabetic patients from type 2. We then wanted to analyze the profile of these human alpha cells that are functional in vivo, and we did a number of analyses, bulk and single-cell RNA-seq, the proteome analysis as well, and very rapidly what we found is that these cells keep a strong alpha cell personality. They have a hybrid profile, alpha-beta, but they are alpha cells basically that are capable of making insulin and secrete it. And all of them are very efficient at reprogramming because all of the alpha cells can become insulin expressors. And because of this hybrid phenotype, we did also a number of immunogenicity tests in vitro, the cytotoxic T cell killing assays, using 2 different clones of T cells from cytotoxic T cells from type 1 patients that were co-cultured with human islets and on human pseudo islets. And basically what we could find is that these native beta cells are more immunogenic than the reprogrammed human alpha cells that make insulin, which appear to be less immunogenic. We also explored the plasticity of other non-beta cells in human islets, and this is for gamma cells. This pseudo-islet of pancreatic polypeptide producing cells, upon transduction with PDX1 and MAFA, we see that they even more efficiently than alpha cells start making insulin after a week, and they secrete it also in response to glucose. And you may think that, well, this is anecdotal because gamma cells represent the minority of islet cells. Yet, this is true only in the pancreas that is derived from the dorsal pancreatic bud, in the body and tail. While in the head of the pancreas, which originates from the ventral pancreatic bud, the islet composition is different, and these islets here are highly enriched in pancreatic polypeptide cells. This is why this plasticity is also very interesting from a therapeutic perspective. And this is why we have analyzed these cells in detail. We have recently published a paper in which we describe the generation of 2 knock-in lines of mice using CRISPR to lineage trace gamma cells, to ablate gamma cells, and to inactivate the PPY gene. And we have been able to characterize the profile of mouse and human gamma cells. We see that these cells are heterogeneous. Interestingly, there are several subpopulations of gamma cells that are bihormonal with different combinations of hormones. We have seen that the inactivation of this gene, PPY, or the ablation of gamma cells has no big impact on glycemia or body weight evolution in these mice. We have lineage traced these gamma cells to a developmental origin, and we have been able to validate in mice what we already found in human gamma cells, which is that these cells can engage insulin production upon massive beta cell loss in mice too. So this cell plasticity is an early discovery. This adaptive functional identity change of differentiated cells in response to injury is an early discovery. what we are trying to do now is a very ambitious systems biology approach, bioinformatics, epigenetics approaches to explore the facultative induction of the adaptive functional plasticity of these cells. We are also exploring pharmacology approaches. We think that it's important to develop delivery tools, for instance, how to target islet cells in situ, maybe with AAV vectors by endoscopy. In any case, I think that these tools that we have now, we can help us to exploit the plasticity to modeling the disease, and think that perhaps it's better to have hybrid cells that make insulin than bona fide native beta cells in a situation, for instance, of autoimmunity. We can also analyze the specific contribution of the non-beta cells to beta cell function. And like I said, we can screen for regulators, chemicals of proliferation and reprogramming of human islet non-beta cells in vitro. So at present, we are doing still a number of developmental studies. We want to define the dynamics of islet cell emergence during development. We are defining the developmental pattern of emergence of the different islet cell types. We want to define the precise postnatal fate of early embryonic hormone expressing cells, and we are also trying to determine in a conclusive way whether there is neogenesis after birth, whether there are progenitors that persist in the postnatal pancreas. And we are doing this with a number of transgenic tools that we have in the lab. To finish, I will mention that we are exploring 4 main areas in the lab at this moment. We are trying to determine the involvement of islet non-beta cells in blood glucose homeostasis. We have mice in which we can destroy the all non-beta cell population. We want to define the mechanisms that lead to diabetes recovery. What is this islet cell plasticity control according to the metabolic status of the mouse? We are also exploring how cell identity and plasticity are regulated in human islets at the gene level, and we are using in particular the CRISPR-Cas9 tool. And we are also trying to develop strategies to induce insulin production in human non-beta cells. Today, for the sake of time, I will just briefly describe these 2 ongoing projects. So with the RIP-DTR mouse, we can, with the amount of the triatoxin that we provide, we can very nicely modeling the degree of beta cell loss, and we can mimic to some extent the natural history of beta cell loss progression during type 1 or during type 2 diabetes. So with this tool, we can modelize the progressive onset of glucose intolerance, hyperglycemia, and full-blown diabetes. For instance, with 2 nanograms of this triatoxin, we have a loss of 1/3 or 1/4 of the beta cell mass. With up to 10 nanograms, we destroy about 50% of the beta cell mass, and then the mice, we see that they are glucose intolerant, and they have a mild non-fasting hyperglycemia. With higher doses, up to 30 nanograms, we can destroy around 80–90% of the beta cell mass. The mice are in this situation clearly intolerant to glucose, and they have over hyperglycemia. And when we destroy the complete beta cell mass, we showed before, we have the very severe glucose intolerance and hyperglycemia. But interestingly, with this tool, what we have here is a way to nicely recapitulate the natural history of, like I said, the progressive loss of the beta cell mass during human diabetes. And in particular, we would like to explore the markers of the initial stages of the beta cell loss in these mice. So what we're doing is, with this approach, to try to profile the whole body and the islet cell responses to this progressive loss of beta cells. We want to determine the dynamics of beta cell loss in diabetes progression. We start studying islet cell hypertrophy, proliferation, reprogramming in response to beta cell loss. We're studying systemic responses to beta cell loss. We're doing the analysis of the metabolic dysregulation. And we are also, of course, trying to understand and define circulating markers of beta cell loss. We are doing a comprehensive blood profiling of a number of metabolites. For instance, in one of these experiments that we are doing to identify biomarkers of beta cell loss and glucose intolerance, we have mice with different degrees of beta cell ablation, and we analyze blood at different moments before ablation, 4 days after ablation, or 10 days after partial ablation, near total or half of the beta cell mass, and so on. And what we're doing in the plasma is to analyze with this multi-omics approach, the lipidomics, the metabolomics. We are doing the proteomics and RNA-seq. We are also analyzing peripheral organs, liver, white adipose, muscle, brain, and doing also lipidomics and metabolomics. And now to finish, we are also trying to identify how to induce production in human non-beta cells. And as you know, cell identity is dictated by a number of conditions, mainly gene expression, but gene expression itself is modulated by metabolism, mainly through epigenetic mechanisms. And also metabolism is key to push the cell into the cell cycle and the cells enter proliferation. So the starting hypothesis here is that metabolism modulates islet cell identity. And we are doing the profiling of human alpha and beta cells. We are doing the multi-omics using several islets of human alpha and human beta cells to profile these cells. And what we want to do with this is to identify the metabolic regulation of gene expression in human alpha and beta cells. We want to do this because we want to introduce modifications in metabolic pathways that will result in alterations in cell identity, because at the end what we want to do is to induce insulin production in alpha cells through this metabolic reprogramming. We want to impose a beta cell metabolic signature to human non-beta cells, and this way we hope to be able to reprogram into insulin production and glucose sensing the human non-beta cell population. And I think that this, if we were successful, this will encompass a reduction in glucagon expression that probably would be beneficial because of the limitation of glucagon signaling in diabetes, and also because these hybrid cells are perhaps, I'm convinced they are indeed, less immunogenic than native beta cells. So just to finish, these adaptive functional changes in cell fates in vivo are now under study, and this is an active area of research in many labs. And ideally, by pharmacology, we should be able one day to promote the body's own cell therapy. So I want to finish by thanking my technicians, also the funding agencies, the European Research Council, the Fonds National Suisse, the Fonds National Clone, and of course all the many collaborators around the world. In particular, I'm grateful to the Juvenile Diabetes because they gave me, by mistake, the first grant. My first grant after I finished my thesis defense was to do this glucagon-free mice. And this was a mistake because they told me later that I was not entitled to receive such a regular grant right after my thesis defense. I want to thank all my fellows, postdocs, and students all over these years. In particular, I want to thank Kenichiro Furuyama, Simona Kera, and Fabrizio Torel because they are extremely brilliant scientists. I'm also very happy with Daniel, Eva, Leon, and Marta that are postdocs in the lab at this moment. And I didn't have the time to mention the studies that I did with Pierre Vasali on the insulitis that is not leading to diabetes, and how activation of lymphocytes is important for the activation of these cells for killing of the beta cells in diabetes. These were very nice experiments. I want to finish by thanking all my mentors, Pierre, Lelio, and Jean-Dominique. They were mentees themselves of Albert Renault, and above all, I want to thank my family. I want to thank my wife and our 3 sons because they are my joy. And this is it from Geneva, the city of the jet d'eau, the water stream. Thank you for your attention.
Thank you very much, Pedro. And I will be a little cheeky and allow a few questions. There are some questions, and I have a burning one, Pedro. So you can push alpha cells, other cells, direction of beta cells. Do you think that non-beta cell islet cells of mice or people with type 1 diabetes who've been sitting for months or years without beta cells in their surroundings and with inflammation around them, do you think they still have this potential to be pushed in the direction of beta cells?
I think yes. Maybe I'm very naive, but I think yes. And one of the reasons is that the necropsies of gold medalists reveal that there are always some remaining beta cells that are dispersed in the pancreas of these patients. The question is whether these cells are escaping the immune system or whether these cells are emerging all the time and being destroyed by the autoimmunity. I think that from what we see here in mice, perhaps the pancreas is all the time trying to make these new insulin-producing cells from the non-beta cells. And I believe so because when we take human alpha cells from type 2 patients also, where there are situations of inflammation, we see that the same plasticity is preserved in these cells and also independently of age. So I'm optimistic about that, and I think that these cells keep this capacity very much.
Excellent. And then there's a question from Catherine Bishop, and she asks, is there potential that these mechanisms, so in the beginning of your talk, can explain transformation of neuroendocrine pancreatic tumors, which convert, for instance, types like glucagonoma to insulinoma?
I think so, yes. I think that this plasticity that leads to regeneration has the same biology behind than cancer, in fact. So yes, indeed, cancer cells are using all the developmental program to try to survive. And what we see in these conditions that we are developing in the lab is that we can push the cell because they have this natural trend to do other things. So a differentiated cell is not something that has identity card in stone forever. Every cell in the body, I'm convinced, needs constant signaling telling them to stop and to remain how they are and doing nothing else that one day know how to do. So my conviction is that yes, they can start doing other things.
Excellent. Then I will just let you read eventually all the very nice comments of people congratulating you, Pedro. And again, I want to congratulate you on behalf of EASD on your work. I want to salute your big heart you have for the scientific community in sharing data, in sharing tools. So thank you very much and congratulations.
Thank you. Thank you for this award. I'm very happy.
And enjoy EASD. Bye-bye, everybody.
Thank you. Bye-bye.